Okay, good evening, good morning to you all. So today, I guess this is the this is the third session for gene technology. So what we will be discussing today is the topic DNA library, right? From DNA library, we'll be starting on, and hopefully, let's see up to which part we can complete by today. Hopefully, we can complete DNA delivery system. Uh, these small sections and then I hope we can complete the analysis part. Yeah, the analysis part. This is a small theory part, and DNA sequencing part. See, right? So by the next class, I'm hoping to complete unit number 7. Right? Today, almost 90%, not 90, 80% of the lessons will be completed. Right? So and then by next class, we'll complete unit number 7, and then we'll move on to unit number 9. Right? That's our plan. So I hope you have revised last week's sessions, right? So last week, what we learned about DNA isolation, which means the separation of DNA, and then we studied about the method called agarose gel electrophoresis. Why we perform agarose gel electrophoresis? To separate the DNA fragments based on their size. That is the main motive. And then we studied about a process called DNA cloning, right? Before DNA cloning, we went to a process which is called DNA probe and hybridization. DNA probe means a single stranded labeled DNA which is used to detect a complementary sequence by hybridization, right? So, and then we went on various, you know, we went to the theory part where we said that probes are prepared by ourselves, and the probes can be labeled using two methods by adding fluorescent molecules or radioactive isotopes, and we can be visualizing them, and they will be transferred to the membrane using southern blotting process. And they are also complete theory part. Last week we clearly learned them, and then we went to the enzymes part. There are three main enzymes. We studied about them. First one was restriction endonuclease. Second one was DNA ligase, and third one was DNA polymerase. And agarose electrolysis is completely done. And then we went on to the DNA cloning, right? So DNA cloning was completed last week. DNA cloning. Recombinant DNA. Yeah, yeah. DNA cloning was discussed. Vectors. So DNA cloning means it is similar to DNA replication machinery. But the problem is here it is done mainly. It is mainly used to copy the desired DNA. It is similar to DNA replication process, but it is taking place within a host cell. That is the thing, right? That is the thing. So for DNA cloning, just inserting the DNA of interest or inserting the DNA which we require, which we prefer into a host cell, is it enough? Means no. Just inserting a DNA which we prefer into a host cell, it is not enough to produce multiple DNA, right? Can you understand? Just inserting a DNA. Now we're taking a organism and we check that this has a DNA which is unique. Now we think that what if we extract the DNA, extract the DNA and insert into another organism? We think about that, right? We just experimentally think, what if? So we are extracting that DNA and we are inserting into this organism. But just by inserting it into the host cell, will it multiply? Means no. It has some set of rules. First one, it should contain an origin of replication for replication, which means origin of replication means the sequence where replication starts off. So to replicate, there should be an origin of replication sequence, right? Or there should be vectors that are required, right? Vectors means they are the vehicles that carries the DNA of interest into the host cell, right? Just like vehicles, they carry the vectors, they carry the DNA of interest into the host cell, right? Okay. And we went on to the vector part. We learned about different types of vectors, plasmids, bacteriophages, yeast, artificial chromosomes. So there we completed. I guess this is the last part we did last week. I guess we completed this, right? And today we'll be starting DNA library, right? Wait a minute. Right. Hmm. Right. We went on, we discussed marker genes, and this is the last. I guess we completed this diagram also last week. So today we'll be starting from DNA library, right? So this is a section where we can understand by just reading it, right? So I'll first read this completely and then explain what they are trying to convey, right? Let's see. When a genome is cut into random pieces by shearing using mechanical force or by restriction enzymes, it creates an extremely large number of different sequences depending on their size of the genome. Okay. Now genome means I have said that there are certain words. Last week I mentioned it in the board also. There are certain set of words. When you come across those words, remember some terms. Can you remember? I said that when you come across the word gene, remember it as a segment of DNA. When you come across the word primary structure, remember amino acid sequence. When you come across the word quaternary tertiary structure, remember it is the functional form of protein. When you come across the word genome means a collection of gene, right? Genome means a collection of gene, right? Okay. Now, okay, collection of gene, right? All these pieces can be integrated into cloning vectors and recombinant can be used to transfer bacteria. Now, with me, you take the tute. If you have the printed copy of the tute, just keep it and just read it with me. Or if you have the screen, no, you have the PDF form. Now I have shared it in the screen. So just with me, you read, right? All these pieces can be integrated into cloning vectors, and the recombinant vectors can be used to transform bacterial hosts, right? These hosts can be cultivated in a suitable medium for selection of transform cells and screening for transform cells with vectors carrying an insert. Since there was no selection for a particular segment of DNA in this case, each transform cell with an insert may carry a different piece of DNA of the genome selected earlier. When all colonies are isolated and cultivated separately, the collection of these colonies is referred to as genomic DNA library. Right? Okay, let's see what they are trying to convey. Right? Now, this is the thing. They are saying, let's take a genome. Right? Wait. What if I change? Right? Let's take a genome. Right? So, genome means genome means a collection of genes. Right? Wait a minute. Do you find any lagging here? I mean, when I'm writing, is there any lagging? Or do you find it as a flow? Is it lagging? It's not lagging. It seems lagging for me. So, okay. Okay, now this is the genome. So they are saying if this genome is undergoing shearing, they're using the word shearing. Shearing means they're saying using a mechanical force, we are separating, we are cutting, we are clustering, we can have various ideas, right? So if we do what happens, they're saying it will be having multiple number of now this is a collection of genes right now. These blue ones are just think they are genes. Now genes means DNA segment, right? Okay, now can you see when we cut this or when we shear this, can you understand that there will be multiple DNA segments like this? Will you agree with me? Yes. Okay. Hmm. So now they are saying that now each of them are DNA segments. Now they are saying, what if we introduce something called vectors? Now this is not new to you all, right? Now you have come across the word called vectors. Vectors means the vehicles that carries the DNA of interest into the host cell, right? Now they are introducing vectors. Vectors are not vector, which means now there are multiple vectors. Now what if they introduce vectors? Now I'll give the shape. This is depicting a vector, right? Now transferring or I'm introducing the vectors into the DNA of interest. These are DNA fragments, right? Now, can you agree that, right? See whether you agree with this point. Can you agree that these vectors, now don't think I'm only introducing vectors. Along with vectors, I can introduce some enzymes called restriction endonuclease, right? DNA ligase. These enzymes also I'm introducing, right? Now, can you agree that when I introduce such, collection of vectors, enzymes, can you agree that these are the structures that will be developed? Wait, I'll complete it. What are these? Hmm. So these are called DNA segments, right? What are these called? What are these called? Give it a try. Hmm. Now here see, red means vector, blue means DNA segments or DNA of interest. Vectors plus DNA of interest. What is called? There's a single word for it. Can you remember? Exactly. Recombinant vector. Very good. Right. Recombinant. Right. Recombinant vector. If I just ask now, sir, how, sir? Now, you said that there was a collection of. No, just think like this. There's a polythene bag. In that polythene bag, just think the polythene bag as a genome. Inside the polythene bag, I have this DNA fragments, which means a collection of genes, right? Now, what I'm doing, I'll open the shopping bag or the polythene bag, and I'm, really releasing those fragments or sequences of DNA, segments of DNA. Now, each of these DNA segments, what I do, I'll be introducing a vector, I'll be introducing restriction endonuclease and DNA ligase. Now, you might ask, sir, why this restriction endonuclease and ligase? I hope you can remember, but still, I'll, recall it, right? I'll remind you. Now, this is the thing. If you see, this is the vector, this is the DNA segment. Just, yeah, give another color. This is the DNA segment, right? Now I have to introduce this into the vector. How can I do? First, I should cut this with the restriction enzyme. I should cut this sequence. And what should I do now? If I cut this sequence, it will be looking like this. Check and tell me whether you agree with this. After cutting, it will be looking like this, right? Right? After cutting that, it will be looking like this. So, and then I'll be inserting this DNA like this. Can I see? But just introducing it, will it join with the vector? Now, see, this is the vector. Now this is the DNA I'm inserting. What I can do is I can bring it closer, but it should join to form a complete circle-like structure. But how can it join? The vector and the DNA of interest. What is the enzyme that joins these two? I hope you can remember that. What enzymes in you have learned many enzymes in a syllabus. What is the enzyme commonly we use for joining purpose? What is the enzyme we use? Just think. Very good. DNA ligase, right? So DNA ligase joins DNA fragments, but now here a vector and a DNA fragment is joined by which bond? What type of bond? Now there are certain bonds right now in our syllabus. Can you remember glycosidic bond? Yes. Glycosidic bonds are present in where? No, where we can find glycosidic bond. Glycosidic bonds are formed when disaccharides are formed. Am I right? When now how disaccharides are formed? Disaccharides are formed by a collection of monosaccharides, right? Two monosaccharides are combining and they undergoing a condensation reaction, right? During the condensation reaction, a water molecule is removed, and this water molecule is removed from OH groups. So now see, normally now you have learned about the chemistry also, right? Now in chemistry you have learned about the phenol structures and those you have learned, right? Now we know that there's a ring like structure for the sugars, right? Like it has some sort of like a ring like structure like this, right? So there will be an OH group here normally, and if you see this is glucose, this is another glucose, right? Now here we are forming maltose, right? Glucose plus glucose, maltose, right? So here also there is an OH group. Now we want to remove a water molecule. So how many hydrogen should be removed? Two hydrogen, one oxygen. So it will be removing like this. And now can you see? uh, you know this, but just I have to remind you this, right? What are the bonds we have learned so far? Now after removing this, can you see the oxygen will be bound like this, the remaining oxygen. Now this oxygen and this structure will be joined by the bond called glycosidic bond. Can you see? Now two monosaccharides are joined by a glycosidic bond. Am I right? Can you agree with that? Yes or no? Yes. Now this is what we learned in monosaccharides, right? The carbohydrate part. Okay. Now that is clear, right? First bond is glycosidic bond. Second one, peptide bond. Where did we learn about peptide bonds? Peptide bond. Where? Can't remember? No, if you can't remember, just let me know. So can't remember. I'll help you to remember it easily. Can you remember where we learned about peptide bonds? Yes or no? Protein, yeah. But if you say specifically, where? Amino acids, right? Between amino acids, what is the bond that is present? Peptide bonds, right? Now, when we form a protein, how many amino acids combine? 20 amino acids. So those amino acids are being joined by a peptide bond. Okay, you're clear with that. Third type of bond is ester bond. Can you remember ester bond? Where did we learn about ester bond? Fats. Can you remember? Triglyceride. Triglyceride where a glycerol molecule and a fatty acid molecule combines to form a triglyceride. So during that we find that there's a bond that joins them which is called ester bond. Now this ester bond is not new to you all because you have learned this in chemistry also. I guess I hope you are learning them. The esterification reaction in organic you will be learning that, right? If you haven't learned it so far, no, until now you haven't learned it in chemistry means you will be learning it in future, right? So fourth type of bond is phosphodiester bond. What is meant by phosphodiester bond? Phosphodiester bonds are present between. Now don't confuse this. In nucleic acids, there are two type of bonds. What are they? Now here in unit number 7, we are learning about DNA, nucleic acids, right? Now here you will be coming across two type of bonds. What are they? One is phosphodiester bond. Other one is hydrogen bond, right? Where can we see phosphodiester bond? Phosphodiester bonds can be noticed or can be seen between nucleotides. Can you understand? Nucleotides. What is meant by nucleotide? Nucleic acid is a polymer. Will you agree with that? Nucleic acid is a biopolymer. So polymers are formed from monomers. Nucleic acid's monomer is nucleotide. So simply this is the structure of a nucleotide. Can you remember? There was a structure like this. This is a nucleotide. Alright? Okay. Where can we see hydrogen bonds? Now this is easy. Where can we see hydrogen bonds? Can you remember adenine, guanine, cytosine, thymine, uracil? Can you remember them? What are they? Nitrogenous bases. So between nitrogenous bases, what is the bond that is present? What is the bond that is present? Hydrogen bonds. Am I right? Yes or no? Yes. Okay. Now I have, shortly, I have reminded you about the bonds also, right? Now back to the topic. Now here, this is the DNA fragment, and the black one is the vector. These two are joined by which type of bond? Phosphodiester bond. Now you might ask, sir, you said that phosphodiester bonds are formed between nucleotides. Now this is between a vector and a DNA, right? I said you that. Now normally, what is the type of circularly what we see vectors we use? So what are the examples of vectors? Plasmid is a vector, right? Will you agree with that? Plasmid is a vector. Yes or no? Yes. Now plasmid, isn't it a circular DNA? Isn't plasmid a circular DNA? Yes, it is a circular DNA. So eventually, indirectly, we can say that there is a phosphodiester bond that is formed between a circular DNA and a DNA fragment. Will you agree with that? Yes. Now, can you agree with that? Can you agree that between a vector and the DNA of interest, phosphodiester bonds are formed? Yes. Okay. Okay, now, so far what you have learned, the genome, if we cut it using a mechanical force or if we shear, what happens? There is a collection of genes that are produced. A collection of genes means a collection of DNA segments. What if now they're saying for these segments or for these DNA segments or genes, what we do is we'll be inserting vectors. Now they haven't said that we'll be inserting restricted endonuclease, we will be inserting DNA ligase. These we have the knowledge of them in the previous lessons. Now this is the thing. When you are going further, when you are going further into the lesson, what happens is the the lesson does not keep on mentioning what we learned before. It won't be spoon feeding each and every information, right? So they will be expecting that the student would have revised the previous sections, so he should have the knowledge of the previous section. So here we should have the knowledge that vectors and DNA fragments are joined by which two types of enzymes. There are two types of enzymes which are involving. What are the two types of enzymes? Just now I said there are two type of enzymes that involve in joining a DNA fragment with a vector. What are the two enzymes? DNA ligase one, very good. Another one. No, that is a bond. That is bond. Yeah, that is the bond. Okay, I'm saying what are the two enzymes? My question was, what are the two enzymes that involves in joining this DNA fragments with the vector? For joining, we can join it using DNA ligase, but we should create a space in this vector for this to be joined. But how can we create it? How can we create this space? by an enzyme. Which enzyme? Which enzyme cuts the unwanted sequence and creates the space for the DNA of insert to be inserted? Restricted endonuclease. Very good. That's it. You understand now? So there are two enzymes involving here. Right? Now this is not a direct point. We should have this knowledge. Now this we learned in previous last class we learned about this. So they won't be again repeating that in this part. Right? Okay. Now they are saying, now what type of vectors have we formed? What type of vectors? Now you just said few minutes ago. What type of vectors are these? Recombinant vectors, right? Right. Now they are saying these are the recombinant vectors, right? I'll take four, right? So there are four recombinant vectors. Hmm. Right. Four recombinant vectors are being produced. Now they are saying, what if we introduced these four recombinant vectors into a host cell? What happens? Just think. Now we have prepared recombinant vectors. They're saying now what if we introduce these vectors into a host cell? What happens? What happens to that host cell? Now I'm expecting a word. If you can't remember, it's no issue because we just learned it last week, right? If you can remember that word, very good because you have got that point. What happens to that host cell? Now say this. I have four host cells. Just I'll draw them, right? Now just think these are the four host cells. Now these are initially what type of cells? They are just normal host cells. But after inserting these recombinant vectors, what happens to those host cells? What happens to those host cells? They get, there's a word. I want that word. They get, can I remember? Can I remember the word? Transformed in last week's class? Can you remember or not? Yes, they get transformed. So transform means simply, right? They'll be inserted into these host cells. So those host cells, what they'll do, they'll be undergoing a process called transformation. Right? They'll be undergoing a process called transformation. And at the end, what happens? The normal host cells will be converted into. Oh, it's a bit lagging here. If you find any difficulties, please let me know. Right. These will be transformed host cells. Right. So transform host cells means simply we should have the idea that they have a host cell plus plus a vector, right? Hostel plus a vector. But the problem is, can you remember last week we learned about marker genes? Yes or no? I hope you can remember. Yes. Now, why did we use marker genes? We used marker genes because they said that transformation is inefficient. Now, within just 5 minutes, I'll just remind what we learned last week about transformation, right? It will be useful because it is almost 1 week, right? We learned that part. So, what I'll do is within 5 minutes, I'll remind you what we learned about transformation in last week class, and I'll correct that with today's class, right? Listen to it carefully. Okay. Okay. Now, the thing last week, what we learned was we said that um, we take a vector, right? And we take a DNA of interest. There's a sequence of DNA, right? Now I have to join the vector and the DNA of interest. But how? Now the vector would be looking like this, right? The vector will be looking like this, and the DNA sequence will be looking like this. In this DNA sequence, I want on this only this sequence, right? So only this sequence is required. So what I'll do is I'll take the restriction endonuclease and I'll cut the DNA sequence and I'll take this DNA and I'll insert here. How I'll insert? I'll first cut this also. So I have cut here and I have cut here. Can you see? I have cut this. Initially, I have cut this segment and I have removed this, and there's a space here. So I have cut this section and inserted. So here we should have knowledge that here and here we have used the same restriction enzyme. Now, if you find any difficulties when you come across the word restriction, restricted nuclease, you're cutting those. Now, just remind that restricted nuclease is similar to a type of scissor. What it does, it will be cutting. It will be specifically cutting a sequence and cuts and joins. Right? DNA ligase is like normal super glue. It will be joining, right? So that is the thing. So after this, what I do, I'll be taking this sequence and I'll join this with the vector, forming what type of vector? Recombinant vector. When a recombinant vector is formed, what I'll do, I'll insert into a host cell, right? When it is inserted into a host cell, what it does is it will be converting a host cell into a what type of host cell? It will be converting a host cell, normal host cell, into a just few minutes ago I said it will be converting a normal host cell into a very good, transformed host cell, right? It will be converted into a transformed host cell. But is this transformed host cells are efficient? Can you answer that? Is the process transformation a efficient process? No. Why? We learned that because for every one transformed host cell, millions and billions of non-transformed host cells are produced. That is one disadvantage. Second one, the transformed host cell, which means the host cells that contains the recombinant vector cannot be distinguished or cannot be separately identified from the non-transformed host cells. Will you agree with that? Now, can you understand what I'm trying? No, I have teach, I taught this last week, right? Now, if you can't understand any words or if I'm going fast, let me know, right? Okay. We cannot distinguish transform hostels from non-transform hostels. Therefore, transformation is inefficient. It is useless, they're saying. Okay, it is useless, it is inefficient. Okay. So what is the we should have solution, right, for this. Now, just by inserting a recombinant vector, if we can't now why we insert that recombinant vector? Can you remember this? See, wait, I'll show the diagram. Ah, see here. Can you remember why we inserted this see this one? This is the recombinant vector. Now, why we inserted this into the host cell? At the end, we expect to form multiple recombinant vectors. Will you agree with that? It's like a business, simple business. I had initially, let's say this is as a 1,000 rupees. I'm in I'm depositing it, or I'm spending that in a method. I'm doing it as some sort of trading or something. Just in a motive, I can multiply it. Can you see? Here we want to multiply it, right? So that multiplying process is called cloning, right? So that was my motive. But they are saying the problem is we can't distinguish அந்த தமிழ்ல சொல்றேன்னு யார்கிட்ட அந்த வெக்டரை கொடுத்துன்னு தெரியும் இப்ப, ரைட்? எந்த ஹாஸ்டல்லு கொடுத்துன்னு தெரியும், ரைட்? Now initially I inserted into hostel, but after inserting it only I'm recognizing that or realizing that I can't distinguish or I can't see to which cell I inserted. That is a problem, right? All will be looking alike. So for identifying transformed host cells from non-transformed host cells, what we do, we'll be introducing marker genes. What type of marker genes? Selectable marker genes. Example, antibiotic resistant genes. We'll be inserting into them and they will be surviving under antibiotic medium. Eh? So they're saying there's another problem. What is it? They're saying even if we give a vector a DNA of interest and we are saying join, will it join means no. Now this is the thing. Can you understand? Now or vector I'll explain it in Tamil. It will be a bit easy to understand. Or vector வச்சு ஒரு DNA of interest எடுத்து ரெண்டையும் join பண்ண வைக்கிறேன். Right? So there is a possibility where we can join it. How? DNA I guess will be coming and joining. But there are instances where the vector will be not agreeing to take that DNA also. Will you now in every possibilities now this is the thing. We should think in now this is the thing. Now we are coming into an exam, right? We are writing an exam. We should expect two possibilities. One is we should pass, and other one is we should fail. There's two possibilities. So similarly, here also there are two possibilities. What is it? One, the vector should take the DNA and form a recombinant vector, or you should reject it. Will you agree with that? Yes or no? Yes or no? Ah, no, no, no issue. That's the thing. Now, simply when we write an exam, there are two possibilities. One, we can pass, one, we can fail. So similarly, here also there are two possibilities. Now what we do, we are taking now, see this is the DNA sequence, right? Now I want only this part. So I'm cutting this section like this and I'm taking that sequence. Now this is the sequence. So this sequence will be taken and this is a vector. Now I want to take this and I cut this space and I'm creating a space. So I'm taking this vector and I'm joining like this. Now I'm giving it here. DNA ligase comes and it starts to join it, right? But the problem is the vector should agree. It should permit. The vector can't. The vector also has two possibilities. One, it can take the DNA of interest and form a recombinant vector, or it can reject. Can you understand? Yes. Now, if there's a question from these sections, don't write that it can reject, it can take. No, those they haven't said. Just for explanation purpose, I'm using this, right? Okay. Now they are saying there are instances where there are vectors with DNA of interest and without DNA of interest. Can you understand that? Yes, there are instances. If it takes DNA of interest, it will be a recombinant vector. If it does not take, there is no DNA of interest. To identify that, we use some other type of marker genes. That was what we learned last week, right? Today we'll be going back to the this part where we will be again coming across this transformation process, right? So here what happens is, here what I said initially will be, let's see what we learned here. Today what we learned was, I said that something called as called genome. Genome is a collection of genes, and I'm cutting it or I'm shearing it, and I'm separating the DNA segments separately. After separating these DNA segments, what I do, I'll be inserting them into vectors, forming what type of vectors? Recombinant vectors. When the recombinant vectors are formed, what I do, I'll be inserting those recombinant vectors into normal host cells so that those normal host cells are converted to transformed host cells. All right. They are saying these transformed host cells will be, now this is the thing, can you see, just try to understand this. They are saying these transformed host cells will be containing identical DNA or non-identical DNA. Because here, see, there are multiple DNA here. We didn't take only one DNA and multiply it. We took several DNA. So these transform host cells, will it contain identical DNAs or non-identical DNAs? Non-identical, very good. You have understood that. Non-identical DNA. Why? Because it was a collection. We didn't no, we didn't check each one of it and we take it. Right? You know, we just separated it out. Or uh simply I can say, yeah. Now this is the thing. I said to you like it was like a polythene bag, right? Now I didn't just put my hand and select each one of them, looking at them, and I didn't pick them. What I did, I completely teared off those polythene bags and I completely released it. And each one of them, irrespective of other, I introduced them into vectors and formed them. So can you understand? Each one of them are non-identical, right? Each transform hostels are non-identical. Now, can you understand? Now they are going to reverse process, right? Now I'll explain it when. Now let's again go back then. Let's see the note part, right? Yeah, where's the note? Here, right? When you come across again the note, you will find there's a bit of comedy here. See, now what happens is they are saying now we have come until the transformed hostile part. They are saying what if we take now they are saying what if we culture this or when we grow this transform host in a suitable medium and when we screen it and we take the DNA segments that are present in these cells, they are saying we can form the genome again. Now, this is the thing they are saying. In each one of them, we have recombinant vectors, right? Can you agree with that? In each one of them, we have recombinant vectors, right? They are saying, what if we separate those recombinant vectors like this and take that DNA out of each of the vectors? We'll be forming the genome again, it seems. Will you agree with that? அந்த ஹாஸ்டலுக்கு இருக்கிற வெக்டர்ல இருக்கிற டிஎன்ஏவை மறுவ எல்லாத்துலயுமே இருந்து செப்பரேட் பண்ணி எடுத்தா we can form the genome again it seems. Yes or no? No? Want to explain it again? You mean you want to explain it again? Oh, you're fine. Fine. If you want, I'll explain it again. Wait. If I draw like this, you will be finding it very easy. Now, can you agree? There is a recombinant vector. The red part means what does this red color denote? Red means see here. Red means what is this red color? See here the this diagram. No, red means see blue means DNA. Red means what is it? See the color. Red means vector. Very good, right? Blue means DNA, right? Can you see now the red one is vector, blue one is DNA. Now what they are saying is they are saying when we take these cells separately, the transform cells separately, and when we screen it out and culture it out separately and sequence that DNA, what happens is they're saying simply this is the idea. If we take this DNA separately, each cell's DNA separately, and when we combine it, they are saying this is called a genome. Can you agree with that? Mm. Right? And they're saying this genome is identical to this, it seems. This genome. Correct or wrong? Correct. Now, this is similar to in chemistry. And have you learned about this type of reaction? What is this type of reaction called? We can say reversible reaction. Yeah, reversible reaction. Now, can you see this is also some sort of reversible reaction? Why? Initially, we are going in the front direction where we cut the genome. We cut the genome and we separate. But again, what they are doing is they are saying, what if we join it again? We can form it to the first, it seems. Can you see? It's a reversible front direction. We went front direction, and they're saying we can, if we go it in the back direction, we'll be forming the uh, reactant back. It seems. Now here, I am comparing the reactant to which structure? See, reactant means the reaction starts off with. So reactant means genome. No, don't mix it up. So in your A level biology, they haven't used those words reactant or reversible reaction. No, no, I just compared it. Right? It is easy for you to understand in that way, rather than just reading on the text that they have given. If you just if you just see the process, it is a bit easy, right? Now let's go to the note part and see whether it is easy, right? Right, right. I missed one point. Now these are transformed cells. So rather than using the word transformed host cells, rather than using the plural host cells, we use a word. Last week I introduced that. Colonies. Colonies of transformed host cell. Will you agree with that? Colonies means what? From one, there's a control. Colonies. Now normally we learn the word colony in our oral history. All history normally we used to come across the word colonies. Sri Lanka was a colony of Dutch. Sri Lanka was a colony of British. Right? So here these are colonies of transform host cell, which means they are cells or they are under control of a genome. Right? Why from genome only we took the DNA fragments, right? Okay. They are saying when these colonies are separated out, colonies are separated out, the collection of these colonies is the one that is called a genomic DNA library. Is that clear? Right? Genomic DNA library. Right? Okay, let's go to the note part. Where is the note? Ah, right here. Right, let's see the note. DNA library. When a genome is cut into random pieces by shearing using mechanical force or by restriction enzyme, it creates an extremely large number of different sequences depending on the size of the genome. Ah, this black, the dark bolded part, depending on the size of genome. So if the size of the genome is larger, what about the DNA segments? You tell me, what happens to the number of DNA segments? Will it be high or will it be low? Larger. Okay. If the size of the genome is very, very small, what happens to the number of DNA segments or what happens to the number of genes? lower. Right? Very good. The first statement is clear, right? This first part. Yes. Now we're going to the second part. All these pieces means what pieces? DNA segments. Right? You're saying the DNA segments. All these pieces can be integrated or joined into cloning vectors, and the recombinant vectors can be used to transform bacterial fossils. Can you see? They are saying all those DNA segments can be integrated or joined with cloning vectors and formed into recombinant vectors, and these recombinant vectors can be used to form transform cells. Is the second statement clear? See here, this diagram. No, rather than my diagram, if you see this diagram, you can understand bit better, I hope. See here, this the line structures they have given here. Can you see this part? Line structures. These are the ones that are present. They are called DNA segments. Right? Now these DNA segments were extracted from where? From where did we extract these DNA segments? Now through this, no, where did we take the DNA segments? DNA segments means gene. From where did we take these genes? Remember that last polythene bag? What is that polythene bag? Genome, right? So from genome, we separated or we took out these DNA segments. These DNA segments we joined with, what are the circle structures? We joined this with, we joined these DNA segments with vectors, and we formed what type of vector? What is this vector called? A vector and a DNA of interest, recombinant vector. And this recombinant vector was inserted into host cell. Now, if you can't keep on typing it, you can use the mic also, right? Host cell. We inserted this into the host cell, and that host cell was converted into what type of host cell? What type of host cell? Transform host cell. Very good. And that transform host cells, what we do? We'll be taking those host cell. Now you should not confuse this, sir. You don't think that like this. Some will confuse that from this host cell we produce these. No, 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 no. Now, with respect to one vector, this is the cell. This is the only cell. So there are there are three more. Those will be combined with vectors and they can form these. Can you understand? For each DNA segment, one vector and one host cell. ഒരു recombinant vector or host cell. Then transform ആയി വരും. Can you understand? Yes. Now, after that, what we do? We perform the function. We perform DNA isolation. Ah, DNA isolation. DNA isolation. Is this word new to you? Is this word new to you? No. The first process we learned under gene technology was DNA isolation. So we perform DNA isolation and we separate those DNA. They will be separating those cells, and those cells are called genomic DNA library. Collection of genomic DNA library. Can you understand? Yes? Right. See, these hosts can then be cultivated in a suitable medium for selection of transform cells and screening from transform cells with vectors carrying an insert. Now they are saying we can separate the transformed cells with vectors from non-transformed cells by cultivating in a suitable medium. That means cultivating means by exposing the cells into a suitable medium, suitable temperature, suitable pH, right? Suitable moisture. Under a suitable environment, if we expose the host cells, right, we can separate the transform host cells from non-transform host cells. It seems, right? Since there was no selection for a particular segment of DNA. There was no particular selection. Now, can you remember this was, I don't know, I can't remember it clearly. In O level, have you learned about the probability lesson? The probability lesson. In probability, there was I can't remember it clearly. There was something, right? Selection and non-selection. Can you remember? Which means uh, the chart. There was a chart like this, right? Something. I can't remember it now. It went on like this, right? So since there was no selection, which means randomly, probability means what? Randomly, right? If there is a selected, probability decreases or something, there was, right? I can't remember it because almost four years since I have done oral maths. I hope there was something like that, right? I don't know whether you can remember it. There was something like that in the orals, right? Okay, whatever it is. Right. Okay. Since there was no selection for a particular segment of DNA, which means I said you right now. I said that the polythene bag, which means the genome. I didn't put my hand and selectively take a gene. What I did was I completely released all of them. I didn't see which one was better, which one was longer, and separately take it. What I did was I randomly released all of them. Can you understand? So as see this, since there was no selection for a particular segment of DNA, in each case, each transformed cell with an insert may carry a different piece. This is a very very important statement. Different piece of DNA of genome selected earlier. Now, I am the examiner. I am asking a question. Why does why does the transformed host cells of the DNA library are genetically non-identical to each other? How would you explain it to me if they ask in a structure paper? Right, I'll repeat the question again. Now I'm asking why, what is the reason for the transformed host cells of the genomic DNA library to be genetically non-identical? So these are genetically non-identical, right? You were the one who said that they were genetically non-identical. So what is the reason? How would you explain it? If you find it difficulty in typing it for a long, you can use the mic also, right? So simply you can answer it, right? They were extracted from a single genome, right? But still, I want a single word, not a single word, a statement. Yeah, they were no genetically non-identical, right? They were extracted from a single genome, okay. But there is another point. What is that point? What did I do to that polythene bag? Now that polythene bag had the DNA in it, DNA segments. How did I release it? Think about the method I released it. How did I release it? Now you say me, how did I release it? Did I look at the polythene bag and separate each one of them separately, or did I release it completely? What I did? Tore the bag and released it freely. So Can you see? Did I select anything? Was there any selection? No, there was no selection. So as there was no selection, what happens? Different fragments of DNA were released. Different fragments of DNA was removed, which means different fragments of DNA was released, and each one of them were non-identical to each other, so that they were inserted into a host cell using a vector to produce a transform host cell, which is genetically non-identical. So exactly this point is the answer. See here, since there was no selection for a particular segment of DNA, each transform cell with the insert may carry a different piece. Different piece of DNA means each one of them were unique genetically, which means they are genetically non-identical. Can you see? Yes. Okay. Last point, when all colonies are isolated and cultivated separately. Colonies means the collection of transform host cells. When all colonies are isolated and cultivated separately, the collection of these colonies are called genomic DNA library. Is that clear? Just go through that part from the DNA library till this part. Just go through and let me know if you have any doubts. Clear? Right. Not yet finished. So now what we can do is we'll be moving to the second type of DNA library, which is called complementary DNA library. Right. So before going over that, there are certain statements. We'll finish it off. Collection of colonies means collection of transform host cells that are produced after cell division of a single transform host cell. Right. DNA libraries. This is a common one. Now DNA libraries are divided into two. What are they? Genomic DNA library. Second one, complementary DNA library. Once again, DNA libraries are divided into two. What are they? Now you tell me. DNA libraries are divided into two. What are they? First one was just be completed. Genomic DNA library. Second one was complementary DNA library. Right? Okay. Now for these, now just few minutes ago we completed genomic DNA library. Now this is a common definition for DNA libraries. Let's see. DNA libraries are collection of microbial cultures, each propagating a different fragment of total genomic DNA, which are cloned in a population of identical vectors. Collection of microbial cultures. Microbial cultures means okay. Now this DNA library part, right? No issue, no issue, right? Now this is the DNA library part. Now this is a general definition, right? DNA libraries are collection of microbial cultures. Now this microbial culture has two meaning. Normally microbes, when you're studying about microorganisms, which is our next lesson, microbiology, uh, we know that microorganisms normally we use culture medium to study them, right? This culture medium means a suitable environment, simply tolerating conditions like suitable pH, suitable moisture, suitable temperature, suitable nutritions, all will be given it as single location, that is called as a microbial culture medium. So here they are using the word microbial culture to denote that DNA libraries are collection of microbial culture, means it is a look, it is DNA library is a collection where we have a completely suitable environment. That is the thing. Can you understand? It is a 100% uh, suitable environment, right, for transformation to be carried out. So microbial cultures means that. Can you understand? Yes or no? Yes. So DNA libraries are collection of microbial cultures, each propagating a different fragment of total genomic DNA which are cloned in a population. Now, I said that there are two meanings, right? So one is that suitable environment. Now, collection of microbial cultures means they are saying the transform host cell. DNA libraries are collection of transform host cells, each propagating a different fragment. They have different fragments of DNA with the total genomic DNA, which are cloned in a population of vectors. They have vectors that are associated with the total genomic DNA, which are different from each other. These collection of cells are called DNA library. Can you understand? So instead of microbial cultures, just put the word transform host cells and read it. Can you understand it? Instead of the word microbial culture, you put the word transform host cells. Don't cut it. Just above, just with a pencil or something, just write it and see. Can you see whether this part is clear? Understood, right? Okay. Inserts. Inserts means Inserts means DNA of interest, which are we are inserting in DNA is called inserts normally. Inserts of each colony. Each colony means each transform host cell can be sequenced separately in order to have a complete sequence of genome. This is what I said as the reversible arrow. Can you see? Inserts of each colony. Inserts of each colony means this one. See, inserts of each colony. This is a colony, this is a colony, this is a colony, and this is a colony. Inserts are this blue color, right? They're saying what they are saying. Inserts of each colony can be sequenced separately in order to have a complete sequence of genome. They're saying if these are sequenced separately, the blue color structures when they are when they are being collected separately, that collection is called a genome. This genome is identical to the initial genome, it seems. Can you agree with this point now? Check and let me know. Yes. Okay. This is how the elucidation of sequence human genome under the human genome project. This is the human genome project that they conducted, right? Early days they conducted a human genome project, and under that they performed something called elucidation. They are saying that that has also a similar idea to this. This is how elucidation was carried out, right? So though they won't be asking those things. So right? Normally in our syllabus they'll be including some information additionally also which are not required for the content, right? Okay. There is another type of now. This was up to the now until this point. With this point, we are coming to the end of uh, genomic DNA library. From here onwards, we'll be studying about complementary DNA library. Right? There is another type of the DNA library which is called as cDNA library, which or complementary DNA library. These libraries contain complementary DNA obtained by reverse transcription of mRNAs isolated from cells. Now, what happens here is now this is the thing. Can you remember transcription? Can you remember transcription? Yes. Where did we come across the word transcription in our syllabus? Can you remember? No. Yeah. DNA packaging. Once we came across the word transcription, but we learned about transcription in protein synthesis, right? In protein synthesis. Now, I have to inform you that I have um prepared a collection of short questions for unit number 7. Now we are keep on doing the content, right? Now we didn't do any papers, we didn't do any questions, right? So I feel that with when we are going each and every class, now it will be this lesson is one of the easiest lesson to forget, actually, right? This is the one of the lessons in the whole our syllabus. This is one of the lesson which is easier to forget. If you didn't practice 1 week, almost 80% of the lesson can be forgotten. If it is 2 week, you won't remember any of the word itself. No, none of the word won't be remembered. So that much of it is easy to forget this content. So what I am planning is I have prepared a set of short questions, not more than 25. Very very short questions, small questions, one word answer, two word answers, maximum one line answers, right? Short questions I have prepared, and hopefully I wanted to inform this in your group also. Now I hope you have seen, I have started for unit 4. Yesterday night I sent a short question set in our group. Did you see it? Unit 4, I sent a unit 4 part which was based on meristems and plant tissue systems, right? Now I'm not compelling or forcing anyone to do it. What from my side I'm trying is, we should keep each and every lesson in hold, right? So what I'm trying to do is, now unit 7 we are doing it, so we should do those papers or questions, right? So there's two options now. Your batch 1 students who are attending on Mondays, they know that we have completed the unit number 7, and we have completed from 2010 to 2025, right? I discussed all the MCQs related to unit number 7, right? Completely with them, we comparing the unit wise knowledge. Now here I discussed only unit 7, unit 7. Now I have given them the structure part also. Now I have completed the MCQ. I didn't get a time to complete the structure essay. I have given the essays them. I have given the essays to them to do it on a daily basis, right? So for, yeah, now for them they are doing it. For you, after this completing, I will be doing that. Past papers will be done, right? But at the same time, I want you to remember this, the lesson, the content, right? The theory part. So what I'll do is I'll be starting. I have prepared. I hope two short questions that I have prepared. So I'll be giving those short questions. The first one will be based on DNA packaging. What you do is just normally on average. Now, just let me know how many hours will it take for you to revise DNA packaging only. If you're revising DNA packaging only, no, how many hours will it take for you to revise it? Just on average, 3, 4 hour or half. Very good. Then you have been now, if it is the first time, it will take almost 2 to 3, right? Why we'll be reading each and every word of them, right? So with practice, very good. From 1 hour to half an hour, you can revise it. So what you do is, after today's class, if you don't have any other works, you just spend half an hour or 1 hour and just revise it. Inshallah, today evening, right? Today evening, I'll be sharing that short question in your groups, DNA packaging. So now I want to ask from you guys. So is it okay if I give the short question on a daily basis or once in 2 day? Oh, do you want it once in per week? Once in per week means the problem is it will be taking a long time. Now I am hoping to complete unit 7 for you all within this month. By next week, I'm trying to complete that, right? And I have to complete the revising also, right? So once in 2 days is good, right? Okay. So today I'll be giving it, and the next one will be given uh, Saturday, right? On Saturday, I'll be giving it. So you just try it. If you have a printer, you just print it out, or you can just keep the PDF form and you can just write the answers on your paper, right? Now, if you have any doubts, what you can do is you just take a picture what you have written the short questions answers, right? You just take a picture of it and send it to me, right? I'll just explain it if you have any doubts, or if you have any errors in writing the answers, right? If I find any no errors, I can point it out. Or if you prefer, sir, I can self mark it. I prefer self marking. You just request me. I can give the scheme also. There are two options. Either the way, I want you to try it, right? Now unit 4 is going on. If you want, you can. But now I know that now at this time you are just coming onto a crash with all the three subjects, biology, physics, chemistry. So it is a bit hard to manage time. I know it. So but still, what to do? We should manage it, right? So from my side, what I'll do is I have prepared the short question part, and inshallah I'll try to give an MCQ paper every week, right? Not more than 30, 20 to 30 MCQs per week, right? I'll give it. And if you want, can do a discussion also, right? Now it's all up to you. If you ask anything, I can give. Even if you want daily basis essays, I can give it, right? So the problem is I can't give a huge load onto you, because it's the first shy for y'all, and you are just going on now, just 4 months left for y'all. So you have to manage it, right? So if I give more excess pressure for y'all, you can't manage chemistry and physics. That's the problem, right? So I'll give it. Works will be given with a small flow, which means now I'm just have started unit number 7 practice, right? So with flow, I'll see the other lessons, your performances, and if you want any revision in other lessons, so I want to I posted a poll in your group also. If you want any guidance or revision in certain lessons, let me know. I can give any MCQ or short question or essays, right? I can give it, right? So please do try that short questions under unit number 7, because this is as I said, this is one of the lessons which can be easily forgotten. So don't forget it. If you forget it now, if it is not in touch for more than 2 to 3 weeks, you have to again revise it from first. That will be taking a huge time, right? So don't let it happen, right? Okay, so today you will be getting that short question part. So take 2 days of time and you can do it leisurely, right? After doing it, you can just inform me. I can give the scheme, or I can correct it and give, either the way you prefer, right? Okay, back to the topic. There is another type of DNA library. This DNA library is called complementary DNA library, right? As I said, we have come across the word called transcription. So in DNA, in protein synthesis, we came across the word transcription. Transcription means transcription, right? Transcription means from a DNA, from a DNA producing a complementary RNA. Now I just tell I have a sequence A, G, C, T. Now this is a single strand of DNA, right? Now I am producing a complementary mRNA for it. How will it look like? Give the complementary strand of mRNA for this DNA strand. How will it look like? Check. Adenine means in RNA it is uracil. Guanine means cytosine. Guanine means cytosine. Cytosine means guanine. Thymine means adenine. Am I right? Am I right? The voice is breaking. Now is it clear, or is it still breaking? Is it still lagging? Now, okay, right? Okay, right. Now, as it was transcription. Transcription means converting a DNA sequence into an mRNA sequence. How? We take a single strand of DNA and produce its complementary mRNA strand. If example, if this is a DNA strand, complementary of it is adenine means uracil, guanine means cytosine, guanine means cytosine, cytosine means guanine, thymine means adenine. Correct, right? Am I right? This is the process of transcription. Is it clear? Yes. Now, how this happens? I taught you in the protein synthesis part. How RNA polymerase comes, strands are separated, and it will be going downstream, right? So, if you see the resource book, they have given a diagram also for that. Now, what we learn here is not about transcription. They are introducing us something called reverse transcription. What is it? What is it? Reverse transcription. Now tell me, if transcription is like this, how will reverse transcription look like? Just think how it look like. No idea? No? No. Transcription means DNA to mRNA. Reverse transcription means mRNA to DNA. That is the difference. Is it clear? Now you might ask, okay, now there we understood that there are RNA polymerase that is coming and those functioning. But here, how does mRNA is converted to DNA? Here also, there is an enzyme that is contributing. That enzyme is called, I'll give another color, reverse trans transcriptase. So here, what is the enzyme that is contributing? RNA polymerase, right? Okay. You might ask, how this, from where does this mRNA come from? Now, normal in cells, there are mRNA, right? mRNA is what? Messenger RNA, right? They are useful in communicating, right? During gene expression, we learned it, right? So, mRNA is useful in communicating, right? So, normally mRNAs are present in cell. So, what we do is, we'll be collecting the collection. We'll be taking the collection of mRNA and separating the collection of mRNA from the cell, right? Can you understand what I'm trying to convey? We'll be taking the mRNAs that are present in the cell and we'll be separating out from the cells. So that separated mRNAs will be collected. That collection of mRNA is called a new word for y'all. What is this word? What is this word? Transcriptome. So transcriptome means what? Collection of mRNAs isolated from cells. Can you understand? Simply, this is, I think, right, clear, right? Now, when I so okay, this is fine. Now, where does the word complementary DNA library come? Now, there we understood what is genomic DNA. Here you are saying about something about mRNA. We didn't come across the word complementary DNA library, right? Let's see what happens, right? See. Okay, right. There is another type of DNA library. These are called complementary DNA library. These libraries contain complementary DNA obtained from reverse transcription of mRNAs isolated from cells or tissues. Collection of mRNAs of a cell is called transcriptome. mRNA is isolated and reverse transcribed into a DNA strand complementary to mRNA. The enzyme used is reverse transcriptase. The second DNA strand is replicated on the first DNA used DNA polymerase to obtain the complement this DNA strand. The DNA fragments are cloned and similar procedures followed as for genomic DNA library to construct a cDNA library. Right? No. Until this point. Now, can you understand until this point? Until this point, from from here to here, is it clear? Don't think about this part. These 3 points, is it clear? Just check and let me know. And then I'll go to the fourth point. Clear. Okay. Let's come to the fourth one. Now, in transcription, now if you can't remember after the class, just revise it, right? And now I said you take half an hour and revise the DNA packaging part. Now, today, if you don't have any other subjects to revise, you can revise it today. DNA packaging, DNA replication can be revised, gene expression, and protein synthesis can also be revised because this is related to protein synthesis, right? You should have some sort of knowledge of protein synthesis to understand this, right? So if you can't and if you can't remember some words, some things, what you do is you revise those sections, and then again, now what the problem is, now you have only 4 months. Now we can't keep on seeing the recordings, right? We don't have sufficient amount of time. So again, revising the sections and seeing the recording, it will be a bit time consuming, right? So maximum what you do is, now after today's class, next class inshallah it will be on Wednesday, right? Next Wednesday. So before that, maximum try to revise those sections. I'm not telling that revise all the sections. Maximum make your effort and try to complete the sections by just revise it, right? And revise and come to the next class, right? That will be very useful. Okay, right. Now that fourth statement, actually what happens is, in transcription, if you remember, what happens is we use only one DNA strand to produce an mRNA. Can you remember? Can you remember that? We used only one. Yes, we used only one DNA strand. So, as we used only one DNA strand, an mRNA strand was produced. So, here, can you see mRNA? Is mRNA double stranded or single stranded? Normally, RNAs are single stranded. So, mRNA is also single stranded. So, mRNA is single stranded. The single stranded mRNA is been reverse transcribed to produce a double stranded DNA or single stranded DNA. Just think this is a RNA, mRNA. Now I'm copying this, or complementary of it is produced, which is a DNA. Will the DNA come as a double stranded one? Like this? Will it come like this? Single, it will be coming as a single stranded DNA. So normally DNA is a double stranded molecule. So they are saying after this DNA is obtained, this single stranded DNA is obtained, what you can do is, they are saying, take this DNA out, it seems. If this is the DNA sequence, they are saying use an enzyme called DNA polymerase. Is the word DNA polymerase new to you all? No. Now, DNA polymerase, where did you come across the word DNA polymerase? DNA polymerase means it is a polymerizing enzyme. So it is useful in producing complementary deoxyribonucleotide, which means producing complementary DNA. So with the help of DNA polymerase, what we do, we will be producing a complementary DNA strand for this. And those two will be winding to produce a complementary DNA molecule like this. like this. Can you understand how a double-stranded DNA molecule was formed? Can you understand? Yes. Right? Okay. They are saying after this DNA is formed, they are saying this DNA should be joined with a vector, it seems. What type of vector? Now you are a bit familiar. What type of vector? I'm sure you will confuse this. I'm sure. Yes, I guessed that. Now, try to understand my question. They are saying this DNA should be joined with a vector, it seems, to produce a to be inserted into a host cell. So DNA is joined with a type of vector to form a recombinant vector. I know I'm sure it will be confusing for you. It's confusing, right? Now this is the now this is a vector, right? What is it? This is a DNA. This is a vector. Now this is a vector. I'm fine, but it has a word before the vector. Here there is a fill in the blanks. I want that fill in the blank word. They are saying this DNA should be joined with the vector to form what type of vector? What type of vector? Recombinant. Yeah, you can understand that, right? You're fine with that. Now, DNA plus vector forming a recombinant vector. But this vector has another name. What is that name? DNA combining with a cloning vector, right? Cloning vector. So before recombining, it is said to be a cloning vector. Can you understand? Yes. A cloning vector with a DNA is called a cloning vector with a DNA is called what type of vector? Clearly listen to the question. A cloning vector with a DNA of interest is called. Now see, this white color, it is a part of a cloning vector with a DNA of interest. So what is this called? Recombinant. Recombinant vector. Is it clear? Is that clear? Yes. Now if you find it difficult because now we have learned it as vector plus DNA is leading to a recombinant vector. If it is difficult for the time being, just ignore it. Don't use the word cloning vector. You are familiar with the word vector, right? They'll be giving marks, right? But even if it is a cloning vector, it's still a recombinant, right? I didn't get the question. You mean this one or this one? Now, cloning vector means this. Cloning vector means it is not yet combined with the DNA, right? Cloning vector is used in cloning process, which means DNA and cloning vector, when it is joined, it is forming a recombinant vector, right? Clear, right? Is it confusing? Clear, right? Okay. Okay. Now what we have formed? We have formed a recombinant vector. They are saying again we can perform the genomic DNA scenario. Can you remember genomic DNA scenario? What? They are also we prepared a recombinant vector. Can you remember? Yes. After that, what we did? Now it is up to you. Tell me. After preparing a recombinant vector, the next step is what? After preparing a recombinant vector, the next step, what is it? Can we just keep that vector in our hands? Can we just keep those recombinant vectors? No, what we should do? Don't panic or hesitate. Just let me know. Even if it is wrong, no issue. You have thought it right. Now you should think in multiple ways. So I want you to think. I won't be scolding or thinking that you have made a mistake. I'm ashamed. No, I won't be thinking those. What I want is I want you to think, right? So now we have prepared. Now just tell me. Now you are a scientist. You have prepared this part, which is the recombinant vector. After preparing this recombinant vector, if you are the scientist, what will you do? Sorry, I'll explain it in time. Use it in a hostel. I know it. You know the answer, right? Now you are a bit hesitating to answer it. Use it in a hostel. Use it in a hostel, correct. But rather than using the word use it, you can use the word inserting. Insert it into a hostel. Can you understand? Now you all have the ideas, but now you are a bit advanced, right? Now advanced level students, but you must use bit advanced terms. So rather than using the word use it in a hostel, we can use inserting it in a hostel. That's it. Right? Okay, very good. Now we have inserted into a hostel. Right? Next step. Right? Next step. This green color is the host cell. Now I have inserted. After inserting this recombinant vector into a host cell, what happens to this host cell? Now you said that you are inserting the recombinant vector into the host cell. Then what happens to that host cell? It is transformed host cell. Very good, very good. And now you are coming onto the track. Now you are using those words. It is a transform host cell. Right, very good. Now, clearly listen to this question. What is the process called when a host cell is converted to a transform host cell? What is that process called? Converting a host cell into a transform host cell. What is this process called? What is it? Don't hesitate. You know, fearlessly answer. Right? Even if it is wrong, no issue. Just think. Okay, host cell. Initially it was a host cell. Now it is transformation. That is the one. Right? That is the process, right? Converting a host cell into a transform host cell is called transformation. How we convert? Now, theoretically, we can say that converting a host cell into a transform host cell is called transformation. But how we convert? We'll be converting it like this. How? We'll be taking the host cell and we'll be inserting the recombinant vector into the host cell so that the host cell will be converted into a transform host cell. Is that clear? Yes, clear. Now you are clear with that, right? Now can you see? Now we have prepared the same scenario which we conducted for genomic DNA library. They are also we prepared this scenario, right? Can you see? This is been repeated again. Okay, let's get back to the part. Now see the fourth statement. The second DNA strand is replicated. Second DNA strand means this is the second DNA strand. This green color one. Now the yellow color one was produced by us. How? What we did? We extracted the mRNA from cells and that mRNA was reverse transcribed into a DNA. That DNA is the one that is given in yellow color. Right? Is that clear? Is that clear? Yes. Now, DNAs are normally double standard. So what we do, we should make it double standard. So what was the enzyme we used to double standard DNA? What is the enzyme we used? We used the enzyme to polymerize and produce a complementary DNA strand. What was the enzyme? DNA polymerase. Very, very good. Right? DNA polymerase is the one that is used to polymerase and produce a complementary DNA strand. Right? Okay, I produced a complementary strand. This is the DNA strand. Right? So here they have used the word, see here, second DNA strand. So second DNA strand means this green color one, which was produced by the DNA polymerase. Can you understand? Yes. Okay, now let's see. The second DNA strand is replicated on the first template DNA. Template DNA means which color one? Yellow color one. Very good. Right? Yeah. Using DNA polymerase to obtain double standard complementary DNA. To produce a complementary DNA. The DNA fragments are cloned. Cloned. Cloned means simply what is meant by cloned. Cloned means, now just in your words try to think what is cloning. Identically multiplied. Very good. You're somewhat there. So here they are saying the DNA fragments are cloned. So DNA fragments are cloned means they have been identically multiplied. How? How they multiplied? They have been inserted into a host cell for multiplication. Can you understand? Outside we can't multiply it. We should multiply it within a host cell. Can you understand? Yes. For that to take place, we need a vehicle. What is that vehicle called? Vector. Very good. Can you understand? Very, very easy, these sections. Right? Students, they are not patient to, connect those dots. Normally, this is like a map. Right? We should connect each and every dots. We have the map like structure, but we are not patient to connect those. Right? Can you see? It is bit connected with everything. Right? DNA fragments are cloned and similar procedures followed for genomic DNA. Now can you see, can you understand a similar procedures followed to a genomic library, this word? Yes. What they are trying to convey is, now this method, this was the same method which we carried out for genomic DNA library also. Right? So they are saying it is similar to that also. Right? Let's see last point. DNA libraries are used. Now they are saying what is the purpose of this DNA library? DNA libraries are basically used as sources of DNA fragments for sequencing. I might ask, so what is sequencing? For the time being, just ignore the word sequencing and go. Because when we are coming further, we'll be coming across the word sequencing. Right? So we'll be coming across the word sequencing. So simply sequencing means, now, yeah, I'll just give an idea of sequencing, right? Sequencing means now identifying the order of nitrogenous bases. Here we have adenine, here guanine, cytosine. So identifying that order, that precise order, that is called as sequencing. Can you understand? Now we'll be going on detail it in for now in the latter part of the lesson. We'll be coming across DNA sequencing also. DNA sequencing is also in our syllabus, right? Here they have just given a hint, right? Can you understand what is sequencing? Sequencing means identifying the precise order of nitrogenous bases, right? Are you clear with that? Yes, right. Okay. What is the purpose of DNA library? DNA libraries are used as sources of DNA fragments for DNA sequencing. cDNA library also reflects the pattern of expression of genes. Okay, now just think practically how DNA libraries are acting as sources of DNA fragments. Now just think it practically. How? Now, just for the sake we can't memorize. How DNA library acts as a source of DNA fragments. Now, just think like this: DNA library is like a storeroom, right? It is looking like a storeroom which has DNA fragments in it. But now, with the knowledge we have learned here, think how. How means now this is the thing. In both the DNA libraries, what was common? In both the DNA libraries, what was common? Joining the DNA fragments with a vector was common. Am I right? Joining the DNA fragments with a cloning vector was common. And forming a recombinant vector was common. That recombinant vector, now this is the thing, this is the beauty. See here, what actually happens here is, see, I'll tell it in Tamil because it'll be easy. ஒரு recombinant vector on produce பண்றோம். And the recombinant vector has the DNA in it, right? The recombinant vector has the DNA in it, but you might ask, sir, why we are inserting it? What is the purpose? To transform a host cell, fine, but why? Now this is a vector, recombinant vector. Why? Now that the question why will be arising throughout this lesson. Why means, now this is the thing. Now if you see, initially we have one vector, one DNA. Can you see? Can you see? Now this is the thing. Yeah, I drew it in the last page, I guess. Wait. Here, right. Now what happens is DNA. They said DNA libraries are sources of fragments. Okay, let's see. Now here DNA fragments were released. Those DNA fragments were joined with vectors, and those vectors were inserted into host cell for transformed host cells, and they converted the host cell into transformed host cell. Why? Why did we did these all these sort of things? Why means we have to preserve the DNA fragment. Now this is the thing. ഒരു ജീനോമില് DNA fragments safe ആയിരിക്കും, right? அதில் இருந்து நாங்கள் வெளியெடுத்து we have preserved them into the transformed cells with a vector. Why? We can use this in cloning procedure to produce multiple DNA fragments. Can you see? இது டெம்பரரியா ஸ்டோர் பண்ணி வைக்கிற இடம் தான் இந்த transformation process. Can you understand? Now you might ask, sir, if it is in the genome, it is better, right? It will be safe there also, right? Is that question arised? Do you have that question? Now, sir, you are saying we have to store that DNA and keep it. But keeping that DNA here, that is also safe, right? It is also present in the genome. Yes, it is safe. But the problem is, does this DNA segments have vector in it? No. For a cloning procedure, vector is essential. So we have to keep vector and the DNA fragments together safely. So if we want to keep those two safely, this is the only method, only safe method. Can you see? Is that clear? We are temporarily storing them and keeping. Why? If there is a need for cloning and producing multi, because all these are identical ones. So non-identical ones. They're unique ones. Now, if they are identical, just by keeping one, it is sufficient. Why? We can produce and multiply them. But as these, each one of them are unique, we can't multiply from one another. Can you understand? Each one of them are unique. So each one of them should be preserved. Now just think all of these are identical. Just for example, I'm saying all of these are identical. If all of these are identical, just think, do we need this much of host cells to preserve that DNA? All have the same DNA. So do we need this much of host cells to preserve them? Yes or no? No, I'm going bit away from the theory part just to explain it. No. Now just for example, I'm saying if these all these DNA identical, Do we need this much of host cells? No. So instead, what we can do, we can take one host cell and keep the DNA preserved. Now you might ask, sir, then how we can perform cloning? When we are performing cloning, what we can do, we can multiply from that only. But the problem here is the word we did not select, as there was no selection. Each one of them are unique. So each one of them are unique. So each one of them should be preserved. We can't lose any of them. We know that genes are very, very precious. So we can't lose any of them. So each one of them should be preserved. At the same time, they should be ready for a cloning. If they are ready for a cloning, they should be associated with a vector. Now this is like an army. Can you see? This is like an army. Genome, you can relate genome with a weapon storeroom. Weapon storeroom. These are weapons, unique weapons, right? Now these are safe in a weapon storeroom, but there should be shooters for that weapons. So vectors are the shooters. So they are temporarily here. The shooters are being kept with the vectors. Sorry, shooters are being joined with the weapons and they are being kept in different camps. That is somewhat you can understand, right? Temporarily they have stored it. Is that clear? That idea? Yes. I hope it's fine. Now, can you understand how DNA libraries are acting as sources of DNA fragments? Can you understand how DNA libraries are functioning as a source? Yes. Is it 100% clear? Because they haven't asked any question from this, which means they haven't asked any essay questions. So they can ask it. So is it clear? 100% is it clear? Let's go through that, right? So just go through that part. Yeah. This part. Yeah. This part and let me know if you have any doubts. No doubts. Let's continue. Just go through this part again. Clear, right? Okay. Then we can move on to the next part. And the next part is bit easy. If you can understand this section, that is very, very easy. Now here, now we are coming to a new topic called DNA delivery system. Very, very, very easy part. Now this is one of the sections where we have been questioned. Now this section will be usually questioned in your FWC papers, term papers, provincial papers. They will be asking if this is one of the sections they prioritize when it comes to exams from unit number 7, right? And but trust me, one of the easiest sections. You can simply understand what they are trying to convey here, right? So DNA delivery, right? From the word itself, you can understand that we have to deliver DNA. How? Can you understand transformation, right? You can understand transformation, right? Transformation means the process where a normal host cell is converted into a normal host cell is converted into a transform host cell, right? That is the normal idea. So during that, what happens? A DNA is being transformed into a, or DNA is converted, or they have been transferred into a host cell. Ah, that is carried out in four methods. Now, the first step is transformation. One is transformation. You have learned this, right? This part, transformation, you have learned. This is not the first time you are coming across the word transformation. Wait a minute. Right. Okay. Now here, this is not new to the transformation process, right? So simply, transformation is one of the type, or it is one of the type through which we can deliver DNA into a host cell. That is the idea. Now, so much of time we spent learning transformation, right? Transformation is one of the type of transferring DNA into a host cell. And there are multiple types. I think you were thinking that transformation is the only way through which DNA can be transferred. Am I right? I hope you would have thought like that. Transformation is the one and only type through which DNA is transferred into a host cell using a vector, right? But not that is not the same. Actually, what happens is transformation is one of the type. There are other types also, right? So transformation is one of those type. Let's see. A cell contacting a foreign DNA is called transform cell. Now this is a definition. Make sure to collect those definitions from unit number 7, right? A cell that is contacting with a foreign DNA is called a transform cell. Transform cell that contains a foreign DNA. Getting the foreign DNA in a cell can be done in using several methods. First one is transformation, transduction, gene gun, agrobacterium mediated gene transfer. So these are the four techniques how we can transfer a foreign DNA into a host cell. Can you understand the concept? Yes, right? Okay, let's go to the transformation part. In this method, a large number of copies of DNA of interest is mixed with host cells. Now here they have put example recombinant vector, which means recombinant vector means what? DNA plus vector, right? So in this method, a large number of copies of DNA are being mixed with host cell, which means they have been inserted into host cells. Correct, right? Now this we have learned it. Now they are explain it again, but in another location, right? Can you understand the first point? Mix means don't think like mixing anything like mixing drinks or something. Those don't connect like that. That mixing here means inserting some sort of, they have, they're trying to convey that knowledge, right? Right. Second one, this is based on the ability of a cell to take up the DNA from its surrounding through the cell membrane, which means, see here, recombinant vector here, this is the recombinant vector they have given. This recombinant vector is present here. There is a bacterial cell. Can you see? Bacterial host cell. So, depending on the ability of the host cell only, even if we give this, it will be taking it. If it has a great ability, it will take. If it is very weak, if the cell is very weak, it won't be taking. So that is what they're trying to convey. This is based on the ability of the cell to take up from the DNA from the surrounding through the cell membrane. Only depending on the ability of the cell, those DNA will be taken in. Can you understand? This is similar to the normal zakat we give. So here there are so many people to give zakat, but there is a set of people, right, who can gain zakat, or who can, yeah. Actually, who can gain zakat, right? There are needy, newly who have been transformed into a Muslim, right? There are orphans, widow. There's a category, right? So based on the ability to take, only we can give. Can you understand? Can you relate with that? Yes. Okay. Third point is the efficiency is very low. Now, this we have learned. We know that the efficiency of transformation is very, very low. There we learned that they said that the efficiency is low. Why? For every transformed cell, millions and billions of non-transformed cells are produced. Second, they said that transformed host cells cannot be distinguished from non-transformed host cells. Right. Now let's see what they tell about inefficiency here. The efficiency is very low in getting the DNA into host cells. Various treatments can increase the competency. There are various treatments to increase the efficiency, it seems, right? So see here, they are not mentioning why the efficiency is low here. Why haven't they mentioned that? Now they have just said that the efficiency is very low. The efficiency of transformation is very low. They haven't given the reasons. Why haven't they given the reasons? Why haven't they given the reasons? Because we have already learned it. If a student does not pay much attention to those sections, and if he comes to learn this part only, will he understand this? No. No, without revising those sections, you can't learn this, right? So just check these 3 points under transformation and we know if it is clear, right? Clearly, each and every statement, patiently look at it, no, each and every statement, and then let me know if it's clear, right? If it's clear, let's move on. Clear, right? Okay. Now, before going on to the transduction process, let me remind you one thing. Now, under vectors, when we learned about vectors, can you remember something we learned called bacteriophages? Can you remember bacteriophages? Yes. So, bacteriophages means what? Virus attacking bacteria, which means simply bacteria who attack viruses, right? Right? Can you remember? I hope you can remember, right? Okay. Now, we said that bacteriophages are also used as vectors. Can you remember? Bacteriophages are also used as vectors. Can you remember how bacteriophages transfer the DNA into the host cell? I used a word. How? I want that word. If you can understand that word, you can understand the further. How does bacteriophages transfer the DNA. Now, normally vectors are used to transfer the DNA into host cell, right? So, bacteriophage is a vector. So, bacteriophages will be transferring the DNA into a host cell through a mechanism. What is that mechanism called? A common word. All of us know it. Even your parents know that. A common word that is used by uh, everyone. What is that? Can you remember? No? Infection. Can you remember? Bacteriophages transfer the DNA into host cell through their infection mechanism. Can you remember? Last class on the board on the on your right side corner, I gave a chart. Physically, I gave a chart and I mentioned that bacteriophages transfer the DNA into the host cell using mechanism. And I gave an arrow downwards under the bacteriophage and I gave infection. Can you remember? Right? Yes. Okay. With that knowledge, let's move on to this. Transduction. This method is based on the ability of bacteriophages to infect host cells, right? Viruses infecting plants and animals can also be used as vectors to deliver foreign DNA into plants and animals, right? Now, don't confuse this with that. You might think bacteriophage is viruses. No, no, no. This is different. This is different. This is based on the ability of bacteriophages. This is depend on viruses or infecting plants and animals also can be used as vector systems like mosquitoes. Mosquitoes are used as vectors to deliver diseases. So similarly here, viruses infecting plants and animals can also be used to deliver foreign DNA into plants and animal hosts. Can you understand that? These 2 points, is that clear? Are these 2 points clear? Just check. If it's clear, drop a message. Clear, right? Okay. Then let's move on to the third point. The gene of interest is integrated or joined into the modified viral genome and packaged into the protein capsid. I'll explain what is that, right? The viral particle can pass the recombined DNA as in its normal infection process. The capsid protects the DNA and this method is more efficient than transformation. Now, this is the thing. What they are saying? They are saying that normally the yeah, this is the if this is the virus, the virus can be used as a vector, right? So the virus contains what type of genome? Viral genome, right? Viral genome is present. So the DNA of interest, if viruses are used as vectors, what happens is the DNA should be inserted into the vector, right? So this viral cell, this is a virus cell, just think, this is a vector, right? So normally we know that vector should be joined with the DNA of interest. So DNA of interest, I'll give the DNA of interest blue color, right? Now this is the DNA of interest. Here, this won't be taken directly into cytoplasm. What happens is it will be joined into the viral genome. You understand? Viral genome means the collection of genes in the virus. Right? See here, viral genome. Is it clear? Is that clear? Yes. So after that, what happens? It will be looking like this. There will be a host cell. Sorry, not a host cell. Virus. This is a vector, right? It has a viral genome. In the viral genome, there are a large number of viral DNA in them, right? Among the viral DNA, what will be present? The blue color. The blue color is the DNA of interest. So now, as a new member is introduced into this, there should be a protection that is required. That protection is given by something called protein capsid. So protein capsid appears like this, like a nuclear envelope, something like nuclear envelope. Normally we learned nucleus, right? About nucleus when we study a nuclear envelope encloses it, right? So similarly here, this protein capsid is present like this, right? Protein capsid. So what does this protein capsid do? Protein capsid protects the DNA of interest during the transferring process. You might ask, "Sir, what is the transferring process?" Ah, what is that transferring process means? Now viruses attacks on what? What are the two things on which virus attack? First on animals, second on plants. So when these try to attack those, this will be directly what will be it will be injecting, right? Normally it will be injecting this DNA into the animal or plant. Can you understand how diseases are caused? Viruses like COVID-19, when they come onto human body, just think as our human body. Now, now this this is the vector. Sorry, yeah, this is a COVID-19 virus, right? This is the human surface. Now, as soon as this comes, can you see this black color? Now, this is the injective point. See, through this, what it does is it will be injecting its viral genome like this. Now, this is the human, eh? This is human, this is COVID-19 virus. So it will be injecting its genome like this, right? It will be injecting, and the virus after some time it will be going away, but the genome will be present in human body. So from the genome it will be producing multiple viruses. Can you understand what happens? Like this only it will be transferring. Can you understand? So as the viral genome is been shared, we have been infected, right? As the viral genome is shared, we have been infected, right? Okay. So as now see, the scientists who found this, they have done a huge study that they have gone a huge study and they have clarified that it is the viral genome that is being transferred. So if we want to insert some DNA, we should insert it into the viral genome. Can you understand? Can you understand? Yes. So through this, they can be introduced so that when it is transferring into animal or plant, this can be, sometimes it can go out or it can be damaged. So what we do, we have been enclosed it with the protein capsid. We are not enclosing it; it is normally enclosed, right? Now let's go to these 2 points and let's see whether it is clear. The gene of interest is integrated in the modified viral genome and packaged into a protein capsid. See here, this is the protein capsid, and viral genome into viral genome, the DNA of interest is inserted. The viral particle can pass the recombined DNA as in its normal infection process. The capsid protects the DNA, and this method is more efficient than transformation. It is more efficient than transformation, it seems. Can you understand this complete transduction process? If it's clear, just drop a message clear. Clear, right? Okay. So we have completed two parts. Now we are going to the third one, right? Okay. This is a small one. Gene gun. In this method, small particles of heavy metal such as gold are coated with large number of copies of DNA of interest, and particles are shot at high velocity into the cells to be transformed. Now, is there is a capsid? How does the infection affect? Yeah, this part, right? Right. You ask, yeah, I mean like this. You're asking the protein capsid is present, sir, here. Then how does the viral genome come out and affect? That is the question, right? Is that question? Yes. Now this protein capsid, what is its function? Protecting the DNA of interest during the transferring process. Am I right? Yes. Now after transferring and going into the host cell, does it have any function? After it is in the human body or plant body, does the protein capsid have any function? No. So will it affect when the viral genome comes out and inserting into the host cell? This is a permeable structure. Don't think it as a rigid structure. It is a permeable. Permeable means it can fluctuate and give space like that structure. So it will be insert. Will it affect like that in the host cell? Will it affect when viral genome comes out? Yes or no? No. See this point? The gene of interest is integrated and packaged like that. That is okay. The viral particle can pass the recombine and normal. See here, the viral particle can pass. They haven't said it will be passing. They have said can pass. Can you see? They are using the word can pass, which means protein capsid permits. Protein capsid permit pannathan viral particle pohil. The viral particle can pass the recombined DNA as its normal infection process. Can you understand? Yes, can you understand? Right. Yes. Now see this diagram. Can you see? If you want, you can just look, give a look at this diagram. The blue means viral DNA. Blue, so yeah, blue means bacterial DNA. Pink means viral DNA. It seems the virus comes and it introduces a viral DNA into it, right? It will be affecting it, causing an infection, right? And through that, what will be forming? Bacteriophages are formed. Now this is not connected. Now have you seen this color, the purple white? Normally this is given by an institute called Byju's. Have you heard about that? In India, they have an institute called Byju's. I don't know whether you have. Do you know something like that? Byju's. Yes, it is an institute, and they have prepared this diagram. Now, this is mainly used for microbiology part, right? It has been used for microbiology part. But what we have to know here is, see this diagram. Not, I don't know whether I can zoom it further. See here, this vector has two types of material, genetic material here. That is what I want to convey. See, test two. Now don't think about these. These are different. See, this bacterial transduction and this different. Right? Can you see this one? This diagram. Now this is a vector. This is a viral vector. Normal viruses get this shape, right? This is a viral vector, right? And the viral vector, what it does? It contains the viral genome, which is pink in color, and it contains the DNA of interest to be inserted, which is the blue color, right? And here see it is introducing both. Can you see? It is not introducing only the blue part. It is introducing the viral genome also, the pink part also. Can you see? Is that clear? It is introducing both. Now can you understand why protein capsid permits? how protein capsid permits. Can you see? Right? Yes. Okay. Then let's move on to the gene gun part, right? Okay. In this method, a small particles of heavy metal like golds are coated with large number of copies of DNA. Interest and particles are shot at high velocity into the cells. Right. Now this is very interesting. What they do is metals such as gold, they make some bullet like structures, metals, right? And normally see here they are coated with large number of copies of DNA. Now when we study this, we think that, see here, when you read this, can you understand that they are saying something like this: heavy metals, gold heavy metals, they are coated with large number of DNA of interest, and they are shot at high velocity into the cells, right? Which means you are thinking that this is a gun-like structure. Those bullets are being mixed with DNA of interest, and they are shot at high velocities. That is the thing, right? That is what you try to just check this, right? Now this is the thing. There will be gold bullets, right? What we do, we'll be mixing it with large number of copies of DNA, and we'll be shooting it with high velocity into the surface. But normally what happens here is it will be creating a small, no, don't think it will be going at a velocity where we'll be, if just think as a leaf, something like a leaf, do you think it will be, tearing the leaf and going? No, these bullets are very, very small, right? They are very thin also, those structures. They can be, yeah, there's a small touch. There's a small damage, which means now if this is a leaf surface, there will be a small damage, minor one, right? Like this. Not a huge one. Wait, I'll show you. It'll be like something like this, right? It'll be looking like something like this. And it will be having a small mark in that. Why we create that mark? To break that cell wall so that this, now this is something like liquid like structure, right? It can quickly enter here, that DNA. You understand? Yes. Just go through this part and let me know if it's clear. This is small part, not much here. This is the equipment that is used to shoot gene gun. Clear, no doubts. Right. Clear, right? So it is agrobacterium gene mediated gene transfer. So this is a bit interesting part, but it is bit longer also. Now we have, it is almost 10:50, right? Shall we have a small break? Because this is a, compared to other three, this is bit longer. You see here, this is a bit long. Up to here, this is not much longer, but still it has bit of content here, right? What we do is we'll have a small break, almost 20-minute break, and then we'll start. Is it okay? Right? It will be a bit refreshing also, right? So it is 10:48. Let's take it is 10:50. So by 11:10, be back. So we can start agrobacterium mediated gene transfer. Right? Okay? Right. Okay then. Uh let's get back to the topic, which is the fourth part coming under the delivery DNA delivery systems. Right now we have learned three types how we can deliver DNA. So first one was transformation, second one was transduction, third one was gene gun. Right now we are coming to the fourth type, which is comparatively different from others. Right, agrobacterium mediated gene transfer. Right, agrobacterium is a soil bacterium. right? Agrobacterium is a soil bacterium which mainly infects plants, right? It mainly infects plants. The mode of infection is very special, right? Wait a minute. Right. Now, first, what I'll do is I'll first completely read it, right? And after reading it, I'll get back to the part of explanation. So it'll be useful for you, which is it will be a bit easy for you, right? Let's see. Now, with me, start reading, right? Agrobacterium is a soil bacterium which infects plants. The mode of infection is very special. The infection causes a tumor on the plant, and the bacterium is living inside it. The disease is called crown gall disease. It is there's a disease that is caused by agrobacterium. That disease is called crown gall disease, right? The cells in the tumor or gall are actually genetically transformed by a segment of plasmid of agrobacterium. I know it is a bit difficult because they are using several terms. Genetically transformed by a segment of plasmid, so they are continuously using that. So it is a bit difficult. I know it is a bit difficult, but let's see, right? Keep on reading it, right? This plasmid is called tumor-inducing plasmid, right? A part of this plasmid can actually be transferred to the plant genome and hence called transfer DNA. Transfer DNA contains genes which induce the formation of tumor and also characterized with pathogenicity, right? What? Yeah, up to this part, right? Now I'll get back to the explanation part, right? Okay. Now what they have said. Now see, Agrobacterium is a soil bacteria. It seems it is living in soil mainly, right? What they do is normally they're infecting animals or plants, mainly plants. They're affecting plants. How? How means they are affecting the plants. They form tumors in the plants, right? Now, when we come across the word tumors, what comes to your mind first as a bio student? What comes to your mind first? Benign tumor, malignant tumor. Am I right? Yes or no? Yes, benign and malignant tumor. But here, the tumor that they speak here is this is related to plants, right? They are saying there are tumors that are formed in plants or galls. Galls, you are a bit familiar. Galls are caused in plants, which you have learned in unit number 2. So they are saying tumors or galls that are caused in plant. They are mainly agrobacterium causes tumors and galls in the plant. How? So that condition where tumors and galls are formed in plants due to agrobacterium, that is called crown gall disease. Can you understand? Crown gall disease means a disease that is caused by agrobacterium in plants due to the formation of galls and tumors. Can you understand? It is an infection actually, right? Can you understand that? Yes or no? Yes or no? Right, right, okay. Right. Agrobacterium is a soil bacterium which causes an infection in plants, right? It is mainly causing tumors and galls in plant. So that type of disease that is caused by agrobacterium is called crown gall disease, which is mainly galls in plants, right? Normal outgrowths. You have seen galls, right? Normally, that something like this in plants they have. If you take this stem, they will have something like outgrowths like this. Like in humans, chicken pox, it is something that take place in plants, right? You can't see the screen sharing. Wait, wait. Now can you see? Right. Okay. Right. It is something like this. If you take this is as the plant's stem, these are called some like galls, right? Normally it can be in plant leaves, right? So outgrowths, mainly we use the word outgrowths, right? Right. Okay. So these are the things that are caused by agrobacteria. So now they're speaking about the structures, right? They're saying that normally in the cells in the tumor, normally this is the thing. Now think this as the tumor that is present in the plant. If you take this tumor, the cells of the tumor are being genetically transformed from a segment of plasmid of agrobacterium. Right. Let's break those words. We know agrobacterium, right? Agrobacterium is a bacteria. Okay. Bacteria have plasmids. Plasmids means what? Plasmids means circular DNA, right? Circular DNA, right? In this circular DNA, they are saying a segment of this circular DNA will be genetically transferred into the cells of the tumor. Can you understand? Can you understand? In the plasmid, a segment of a plasmid will be genetically transformed into the plasmid. Sorry, into the cells of the tumor. Can you understand? Yes or no? Is it clear? In the plasmid, a segment of it will be genetically transformed into the uh, cells of the tumor, right? Okay. So that plasmid, right? Those plasmids are called tumor-inducing plasmid. So what is meant by tumor-inducing plasmid? Tumor-inducing plasmids are the plasmids that have segments which develop into the cells of the tumor. Can you understand? I'll repeat it again. What is meant now? Here, see, they have just meant as tumor-inducing plasmid. So I'll give a definition also. You right? Don't write it in. They won't be asking because they haven't given a definition. So I'll just give it for your understanding purpose, right? So what is meant by tumor-inducing plasmid? Tumor-inducing. Now see, Ti means T means tumor. I means inducing. Ti plasmid, right? Tumor-inducing plasmid means the plasmid that has a segment that will genetically transform and develop into a cell in the tumor. Can you understand? Is that clear? Yes. Sure, right? Okay. Now, what happens is the bacteria will be living inside the, actually, it will be living inside these tumor cells. Now, it will be like a dome shaped structure. Just think this in a 3D shape, this, right? As a dome shaped structure, and the bacteria will be living inside it. That is how the bacteria survives, right? Can you understand? That is how the bacteria survives, right? Okay. A part of this plasmid can actually be transferred to the plant genome and hence called transfer DNA. To explain this, I'll explain like this right now. Now see, what is this red color bump? This red color bump, what is that called? Tell me that. No, the red color one? In a stem, those bumps or growths that take place, what is that called? In unit 2, you have learned. What is that? Tumor, goals, you can say whatever you want, right? But they have said it as tumor, so you better use the word tumor. So this is a tumor. Just think this is as a stem, right? This is a stem. This is the tumor that is being created. Can you understand? Can you connect it? This is a stem. Now, have you seen normally there are some plants where in the stems they have like this structures? Have you seen? some have in their leaves, right? So similarly here, this is the stem. In the stem I have drawn it bit enlarged. This is one tumor. Can you understand? Yes or no? Yes. Okay. So this is a tumor, and these small, what are these small structures that I have drawn? Cells of the tumor. Correct? They are the cells of the tumor. Yes, exactly. Cells of the tumor. So these cells of the tumor are being, now we are saying that these are cells of tumor, but actually what are they? Actually what are they? They are the, tell with me now, they are the segments of plasmid. What type of plasmid? What type of plasmid? They are segments of, there's a word, what type of plasmid? Just few minutes ago we learned. They are segments of what type of plasmid? Tumor inducing plasmid. Is that clear? Check again, check. See here. The cells in the tumor, these are actually genetically transformed by a segment of plasmid. See here, agrobacterium. Okay, I'll repeat it again, right? See here, agrobacterium has plasmids. Those plasmids are circular in nature, they are circular DNAs, and those circular DNAs have segments which can genetically, not all the segments, there are specific segments which can genetically transform into cells of the tumor. Cells of the tumor. Those plasmids are called tumor-inducing plasmids, right? So, what is meant by tumor-inducing plasmid? Listen carefully. Tumor-inducing plasmid means the plasmid that has segments that can, you see the words that I use, that can genetically transform into cells of tumor in plants, right? Now this part, now what they are trying to tell is, see here, this is a stem. Just think as this is a stem, right? Now this is a tumor. In the tumor, these small structures are what? These small structures are cells of the tumor, right? They are cells of the tumor. But now we are saying those are cells of the tumor. But actually, what are they? Genetically, what are they? Just now I said, genetically these cells are, what are they? See the structure and see, see the structure and let me know. Genetically, these cells are, is it hard to understand? If it is hard, I'll use another technique, something, and I'll try to explain. You want me to explain it again? You want me to explain it again? Yes or no? Yes, okay. Right, I'll explain it again from the first, right? No issue. Now, this is the thing. Now, agrobacterium means a bacteria that is present in soil, right? So, what happens is this bacteria is present in the soil, and it is mainly infecting plants, mainly plants, right? How? It will be affecting the plants by an infection called crown gall disease. Crown gall disease, sir. Okay, what is meant by crown gall disease? Crown gall disease means this agrobacterium infects the plant by causing tumors in the plant. Okay, so why does it form tumors in the plant? Because it forms the tumors in the plant and it lives inside the tumor. Can you understand? It will be forming tumors in the plant and it will be living inside the tumor itself. See, the cells in the tumor or gall actually, no, this one. See, and the bacteria lives inside it. Infection causes a tumor on the plant and the bacterium lives inside it. So it will be forming tumors. Why? To live inside the plant, to live inside the tumor and be hidden, right? It want to be in contact with the plant, but at the same time it should not be exposed. So how can it live? It can live only inside the tumor. Can you understand? Inside the tumor. Is that clear? Clear. Very good. Right? Right. Now we are coming to a genetical part. What means now? See, this is a stem. Right? Just take this as a stem, and this is a tumor. Is this diagram clear to you? Or is it hard? Or should I draw another diagram? If you want me to draw another diagram and explain, I'll explain. It's clear, right? Okay. Now just think this as a stem, and this is a tumor. Now, tumors are made up of cells. We know that from our Unit 2 knowledge. So, this is a tumor. What are these small structures? They are the cells of the tumor. Right? So, when we see it actually from like this, we say that these are cells of tumor. But actually, genetically, right? Genetically, what are they? Genetically, let's see. Genetically, what me? Now, genetically, this is the thing. Agrobacterium has plasmids. Why? Because agrobacteria is a bacteria. Normally, bacteria, prokaryotes have plasmids. Do you agree with that? Normally, prokaryotes have plasmids. Yeah, we have learned that in DNA packaging as well. So they have plasmids. Plasmids means circular DNAs. They are circular in nature, right? Those circular DNAs have DNA segments. Listen carefully. They have DNA segments which can, right, which can genetically transform into the cells of the tumor. They will be disguising. Just think like that. They are disguising or they are developing into the cells of the tumor. Can you understand? Right? Now, now tell me that whether this gall or this tumor is it. Now when we look it from outside, how will it look like? No, normally I don't know whether I have in your resource book, do you have a picture of? No, right? You don't have a picture, right? Wait a minute. Give me a second. On I'll show you a picture so it will be clear for you, right? Right. Wait, I'll share it. Can you see the picture? Can you see this picture? Yes, right. Now, can you see this is the stem, right? Now, this is a tumor. Now, externally, when we see this, what we think is the plant stem is present due to some reason, due to disease, the plant stem parts have swollen. That is what we think. Am I right? Normally, we would think like that. Yes or no? Yes. But actually, what is me? This is actually, wait, these, see here, these are agrobacterium, right? These are the tumors that are formed. So these tumors have cells in them. Those cells are, how are they formed? Those cells are actually genetically transformed from tumor inducing plasmid. Now you have the picture right now. With this picture, let's move on to the note, right? Clearly see this picture and then we'll move on to the part, right? Wait, let's move to the other part. right? Can you see the screen? The note part. Do you see the note screen or other? No, right? Wait. Mm, wait. Now, can right? Yes, okay. Right. Now you can remember the picture, right, which I showed now. Right. With that, let's go. Now, the structures which I showed you, those bump structures, they are the tumors. Internally, they have cells. Those cells are actually formed by the segments of the agrobacterium plasmids. I'll explain it, Tamil. ഇത്ര നടക്കുന്നത് അക്രോബാക്ടീരിയമുക്ക് പ്ലാസ്മിഡ്സ് ഇരിക്കുന്നത് right. Plasmid and a circular DNA. And the circular DNA, there are segments of DNA which can develop or genetically transform into the cells. So eventually, those segments of the tumor-inducing plasmid are the ones that have been formed into tumor cells. Can you understand? So Agrobacterium has given its own segments of tumor-inducing plasmid to cover itself and live inside. Can you see? Agrobacterium has given its own segments of tumor-inducing plasmids to form tumor cells, and after tumor cells are formed, agrobacterium lives inside it. Can you understand? Is that clear? Yes. Right. So now check this point. Is that clear? 100%. No doubt, right? Even if you are a bit confused, let me know. I'll explain it again. No, you mean you have a doubt or you don't have any doubts? You want me to explain it again? No doubts. Okay. Then let's move this part. A part of this plasmid can actually be transferred to the plant. See, they are speaking about the plasmid twice. First, why they spoke about plasmid? To relate it with the cells of the tumor, right? To relate with the cells of the tumor. Now they are saying a part of the plasmid can actually be transferred to the plant genome, and it is called transfer DNA. This is easy to understand. Why? Now this is the thing. Now think like this. The agrobacterium is the yellow color one. Agrobacterium has plasmid in it, right? I'll give a blue color, right? See, the blue color is the circle plasmid, right? A part of this plasmid can be genetically transferred to the plant genome, which means now see, this is the plant stem. The plant stem is a part of the plant, right? So it has the plant genome in it. I'll give green color for the plant genome, right? Plant genome means the collection of gene. This is the plant genome. So a part of the plasmid of the agrobacterium is being transferred into the plant genome. That DNA is called transfer DNA. I'll explain it again, right? Listen carefully. Now, agrobacterium. Wait. Now, see the screen. Wait. Right. Right. See my screen, which I try to explain. Right. Now, see, if this is the plant stem, right? If this is the plant stem, I don't have a stopwatch to explain. Right. Okay. Now this is a plant stem. Just think in a circular, there is a goal, right? There is a tumor that is formed, right? Now inside the tumor, the agrobacterium is living. Is that clear? Yes. Now you might ask, how did the agrobacterium form that tumor? What it did was it gave its own plasmid, its plasmids, some segments. It sacrificed its own plasmid segments to convert those segments into cells, which means agrobacterium have now we until now what we have learned just plasmids, right? Agrobacteria have some special type of plasmids called tumor inducing plasmid. Is that clear? Agrobacterium have a special type of plasmids called tumor inducing plasmid. Yes. What those plasmids do is they have certain segments which can develop into tumor cells. Tumor inducing plasmids have special segments that can develop into the tumor cells, and they will be completely forming a tumor, complete wall like structure, a dome shaped structure. Is that clear? I know it's a bit difficult to connect it because you should relate it or try to understand it in a 3D manner, right? So it's a bit difficult. Wait, what I'll do is it's a bit lagging here. That is the only problem. If not, I can explain it. This is very very important, right? Okay, I'll explain it as a story. Then it is a bit easy, right? What is this? This is the agrobacterium. Okay, okay. No, no, wait. I'll give another shape rather than using the circular shape because plasmid is circular, right? So I'll give a box shape, right? This is agrobacterium. Now the agrobacterium chooses this plant to be affected. Now it comes. Now, just as it comes to the plant, it can't survive in the plant. What it should do, it should live within the plant with with the plant's contact. It should be in contact with the plant to affect it. So what should it do? It should temporarily form a residence for it. You understand? Yes, it should temporarily form a residence with it. So what it does, what is its residence? If you are agrobacterium, what you will do to stay with the plant? What are the conditions? You should not be exposed, but you should be in contact with the plant. The only choice is forming a tumor. Am I right? Yes, tumor cell. No, tumor. Just mention it as tumor, right? I'll come to the tumor cells part. The only choice is tumor. Why? If I form a tumor, I am not exposed. You might ask, why? Because I am living within the tumor, not on the tumor. As I am within the tumor, if just think I am the agrobacterium, as I am within the tumor, I am not exposed to the external environment. So I am escaped from the external environment. First. Second one is still I am in contact with the plant. I am in contact with my target. Can you understand? Yes. Is that clear? For going further. Yes, clear, right? Right. Now this is the agrobacterium, and agrobacterium contains some special type of plasmid. This blue color is the one that is called what type of plasmid? Tell me what type of plasmid? This blue color. The special type of plasmids that are present in agrobacterium are called what type of plasmid? TI, tumor inducing plasmid. In this tumor inducing plasmid, you might ask why I haven't completed the structure. Wait. There are special segments that are called, this green color is the segment. There are special segments that are called, yeah, not called, they are special segments, right? Okay. Now, can you understand this? The red color is the agrobacterium, the blue color is the tumor inducing plasmid. In the tumor inducing plasmid, there are special segments, green color. Is that clear? Yes. Now, what happens is it has to form the residence, right? Now, it can't ask help from the plant. Why? because it has come to kill the plant. It has come to affect the plant. How can it ask help for the plant? So what it should do is it should make a sacrifice. So it will be giving its own plasmid segment to develop a residence. Can you understand? So it will be using this green color. It will be it will be removing and it will be giving this green color, and this green color will be genetically transforming into cells. It will be appearing like this. No, I can't keep on drawing, so I draw like this. Right? Is it okay? You can understand, right? I hope it is clear. Not spring, those are cells, right? I can't keep on drawing them, so I have drawn like this. So just imagine there are multiple cells. Clear? Is that clear? Yes. Now, now can you understand that? Right. Now let's see. Can we explain that statement? Where is that statement? See. The cells in the tumor are actually genetically transformed by a segment of agrobacterium. See here. The cells in the tumor are actually genetically transformed by the segment of the plasmid of agrobacterium. Blue is the plasmid, green is the segment. So this blue color plasmid is called tumor-inducing plasmid. Why? No, you might ask, sir, why? Because the plasmid does not do anything, no, sir. The green color structure is the one that is doing. Yes, but still, as this plasmid is the one that has that segment which can form into cells, that plasmid is called tumor-inducing plasmid. So this blue is the one that is called tumor-inducing plasmid, not the green one. This blue one is the one that is called tumor-inducing plasmid. Is that clear? Sure, right? Now see, agrobacteria, the red color is living within the cells. Can you see? Is that clear? Yes, right? No doubts, right? No, don't hesitate to ask. Even if it's still, it is, if you can't understand, let me know. I'll explain. Don't move on with, confusions or doubts. Clear, right? Right, very good. Right. I'll just increase the width of the stem, right? So that I can explain it a bit clearly. Right. I'll do it something like this, right? Okay. Now I'll move to the next part. Now there is another segment like this, the plasmid. Green one was what is this green color called? Now explain it to me. What is that green color? I'll explain the yellow color one. You explain the green color to me. What is that green color? Just give a try. No issue if it's wrong because it is the first time you're coming across this, right? So just give a try. What is this green color? Segment of plasmid. Very good. What it does? If you can't keep on typing, you can use the mic also. It transforms into, now here you have you done it correctly, right? You have written it correctly. It transforms into cells, but you have to use one term. It genetically transforms, right? It can't directly transform, it will be genetically transforming itself, right? So it genetically transform into the cells of the tumor. Very good, right? Okay. Then just because of that segment is present, what is that blue color one called? Tumor inducing plasmid. Very good. Right. Now I'll explain what is yellow. Yellow color. There's another segment in that. Now what it does is a part of this segment will be genetically, see, it will be actually transferred to the plant genome. What is that? Now what happens here is, I'll give a white color. Now this is the plant stem, right? Plant stem has plant genome. Will you agree with that? Plant genome means a collection where plant genes are present. Correct, right? It has a plant genome, right? This white color, the green cells are actually tumor cells. Yes, green cells are tumor cells, right? Any doubts? No, right. Okay, perfect. The white one is plant genome, right? So what now happens is this yellow color segment, a part of this segment will be transferred, not transformed. Initially, we said that it is transformed. Now it is transferred into the plant genome, it seems. Can you understand? A part of the segment is transferred to the plant genome. It is a bit confusing. It is transferred to the plant genome, it seems. This is called, now this is called transfer DNA. Now you ask, so it is a segment. How come you say it is a transfer DNA? Correct, right? Now this is a plasmid. Plasmid is circular DNA. We are using the word segments to denote that it is a DNA, a collection of DNA, right? DNA segments, right? Now, can you understand this yellow color is the one that is called transfer DNA? This green color, this is also a segment of DNA. Will you agree with me? It is also a segment of DNA. Yes or no? Yes. Right. Now, this yellow color one is called transfer DNA. What happened? Now, explain to me what happens to the yellow color one. What actually happens to the yellow color one? It combines with the gene of. Yeah. Rather than using the word combine, better use the word transferred. It is being transferred to the. Combines means what? Combines means both two things should be mutually joining. Here the plant genome remains the same. It does not want to join. It is just present. This is the one that is forcefully coming and joining. So it is transferred, right? A part of the segment is transferred to the plant genome. Correct? Fine with that? Transferred into the plant genome. Very good. Right? Okay? Yes. This yellow color part is called, what is a special name we give to it? tDNA. tDNA. Very good. Right? Very good. Transfer DNA. tDNA means transfer DNA. Very good. Now let's go to the note part. Now you might ask, why is it transferred? Let's see. Let's see. With the note, I'll compare because this is very, you should have. Now see, they have given a diagram. Can you see? Now this and this are somewhat similar right now. Here I have given two. There are multiple ones. See here, see. There are some other structures also. Can you see? Now these are not required. For our syllabus, we need only two of them. Right? Can you see? Not two, three. There is another one. Let's see. Right? Okay. Transfer DNA contains genes which induce the formation of tumor and also characters associated with the pathogenicity. Very right. Now can you see? Can you somewhat connect what happens actually? Now, now be honest. Tell me, did didn't you think like this? How come this green color? Initially I said that, but didn't you think how come a DNA develop into this much of cells? Didn't you think like that? Yes, right. Now, can you see here? It is not that segment only. It can. Transfer DNA contains genes that involves in inducing the formation of tumor. Can you see? There is a supporting character also. Can you see? There is a supporting character also. And also characters, now it is inducing the formation of tumor and also induces pathogenicity. Pathogenicity means it is the reason. Now this yellow color is the one that is the reason why agrobacterium is pathogenic. If we remove this yellow color, what happens? Now just think this yellow color, it is the one that is causing the red to be pathogenic, right? Correct? Will you agree with that? Transfer DNA, it is inducing the pathogenicity, right? Wait, will yeah, we'll move to the next part and I'll explain it again. What is needed for the transfer of DNA is right and left border sequence of transfer DNA. Right and left border sequence of transfer DNA. Let's see. Wait. Now, see here. There are left. Yeah, see here. The yellow color one is the transfer DNA and it contains left transfer DNA, right transfer DNA. There are two sequences. Now simply this is the thing. Here also they have given yellow. In my structure also I have given the yellow. Now simply what is they're trying to convey is this transfer DNA to be converted into the or transferred into the plant genome. It requires two sequence. It seems first one right border sequences and left border sequence. There are two sequences that should be present. Correct? Is it clear? Yes. Right. Okay. Therefore, scientists have removed most of the bacterial genes, including virulent genes for the transfer DNA, and have made room for the inserting the genes of certain border sequences. Right? Right. Now, just read this. Oh, no, no need. I'll explain. Right? Now, actually, until now, what we have learned DNA delivery system, which means the methods how we can insert the DNA we wish. Right? Is it clear? Until the gene gun, in all those three things we were clear that we can select the DNA what we want. In transformation we understood that the DNA we want, we inserted it with the vector and we inserted into the host cell. In transduction, the DNA we want, we inserted into the viral genome using a protein capsid. In gene gun, what we did, we mixed copies of DNA with the bullet and we shot at high velocity to the host cell. In these three things, can you agree that we were the one who chose the DNA we want? Yes. Now here, can you see, are there any human involvement here? It is the agrobacterium that is doing itself, right? Now you might see, sir, this is an infection process. Why did they use this method for the DNA delivery system? Can you understand? I know that multiple questions now it comes on your mind because now we were going on a flow. You are coming, so you are saying again, you are giving another doubt, right? And I feel it. I can understand it's a bit difficult, right? Can you understand that? Don't get panic. I'll explain it clearly, right? Just answer whether can you understand that there is no human involvement here. Until this portion, there is no human involvement. Can you understand? Yes. So what happens is, right? Now I'll draw the structure bit enlarged, right? For explanation, right? Don't panic. The same diagram, but bit enlarged. So the green one. What is this green color one? Now when I'm drawing, you just tell to yourself the green one is a segment of plasmid. What type of plasmid? Is it normal plasmid? Is it normal plasmid? Tumor inducing plasmid. Very good. Right. This yellow color. What is this yellow color called? Segment of DNA. Yeah. But it has a special name. What is that? Transfer DNA. Clear? Now, okay. Transfer DNA. Right. Now, this transfer DNA contains a right. Now, see, just if we take it this manner, you can see that it is a right sequence. This is the left border sequence. Any difficulty in that? Understanding in that? Any difficulties in understanding that? No, right. Now, they said that this yellow color one, what it does, this transfer DNA has right and left border sequences. And what is this function of this? Transfer DNA induces what? Induces tumor formation. And it controls pathogenicity. The second one, it controls pathogenicity. Okay. If now clearly understand that if anything controls pathogenicity, it has something called virulent genes. Now don't panic, because you don't think the answer. Again, a DNA segment. Why? Yeah, this is called as virulent genes. Now, don't think these are separate. Transfer DNA contains virulent genes. Is it clear? Virulent. Wait a minute. This is a bit lagging. Right. Virulent genes. Right. What this virulent genes does is it is the one that is responsible for pathogenicity. Can you understand? Can you understand? Yes. So, as transfer DNA, the yellow color one, the transfer DNA contains virulent genes in them. Transfer DNA is responsible or induces pathogenicity. Can you understand? Virulent genes. Oh, I know it is a bit difficult. Listen carefully. Virulent genes are the ones that are useful, or that are virulent genes are the ones that causes diseases. Virulent genes are the one that causes diseases. So you can say that because as transfer DNA contain virulent genes, transfer DNA induces the pathogenicity, which means in the virulent gene ikira suttitan in the transfer DNA no help parapudu. It causes pathogenicity, induces pathogenicity. Can you see? Actually, virulent genes are the one that is causing diseases. Transfer DNA does not do anything. But when we see it externally, we say that transfer DNA is the one who causes or induces pathogenicity. Now, this is the thing. You have a friend group, right? You have a friend group where you have three friends. Among those three friends, one friend, right? One friend is not studying in your class. You three are sitting together. The one who is not studying is the one who is making noises, who is not doing the work, right? Suddenly sir comes or teacher comes and checks her book, and finds that she or he finds that she has not done the work for the past 3 to 4 months, and she has found that she's not doing anything, and she have found that uh, she's missing classes, she's not coming to the class regular, and she comes to know about her a bit deeper. Now, automatically, she, without knowing, now you both do the work, right? Automatically, she thinks that you two also have some sort of characteristics similar to this girl. Correct or wrong? Whenever she again teaches in the board, whenever she looks at you three, she will be looking like you three don't study. Just because of that one student, you both also get affected. You both also get the name. you are not studying, you are lazy, those stuff. Can you understand? Just because of that one. Right? This normally happens in classes, right? Just because of one student, the ones who sit near them gets that blame, right? So similarly here, transfer DNA contains virulent genes in them, right? But virulent genes is the one that is involving in causing diseases. But when we externally see that, we say that transfer DNA is the one inducing pathogenicity. But actually, who causes that pathogenicity? Actually, who causes that pathogenicity? Virulent genes. But who gets the blame? Transfer DNA. Can you understand? Yes, right. Okay. Now, scientists understood that. Can you understand here? Now, this is like a flow chart, right? Now, can you understand? See here. Now, I'll explain it again with another diagram, right? It's a lots of work, but it is a bit interesting, right? Pay your attention. Last part, right? Right. Now, let's see. Now you might understand what how actually now here I gave it just right a diagram to explain it each and every. Now you will understand stepwise. Now the agrobacterium comes. This is the plant structure which it aimed for. It is coming here, right? It contains a plasmid. What type of plasmid? What type of plasmid? Tumor inducing plasmid. Very good, right? It contains this yellow color one. What is this yellow one called? A segment of plasmid. What does this green color one do? A segment of plasmid. Very good. What does this segment of plasmid do? Form tumor cells, right? It will be genetically transforming into tumor cells. Very good, right? Now we have the yellow color one. What is this yellow color one called? Segment of DNA, right? But it has a special name. What is that special name? Transfer DNA. Very good. Right? So when you mention this green color, you say it as segment of DNA. Fine, because they haven't given a special name. But here they have given a special name which is called transfer DNA. Right? It is also segment of DNA. Correct. But they have given a special name, right? So as they have given a special name, we have to use that special name. It is transfer DNA. Clear, right? I hope it's clear. Clear? No doubts, right? Yes. Okay. Right. Which color did I give? Red color. Okay, I'll give the red color back again here. I'll give this red color. What is this red color? Virulent genes. Very good. Right? Okay. Now let's see what is it Azan? Let's wait for 1 meter. It's Azan, right? Right. Right. Okay. Now, right. Now this is fine, right? Now the red color part is agrobacterium. The blue color part is the tumor-inducing plasmid. The green color part is the segment of DNA which will be genetically transformed into the tumor cells. The yellow part is the transfer DNA. Red color part is the virulent genes. Clear, right? Yes. Right. I'll include another two main important parts. This and this are right and left border sequences. Clear, right? No doubts. Clear? Ah, yes. Okay. Now let's see how the tumor cells actually form. Now they haven't given those in steps, but you have to understand it as a story, right? Right. Let's see how it actually forms. Now, what does actually how does no what causes the tumor cells to be formed? This green color is the one that is genetically transformed. But what induces the tumor cells formation? Let's see here. Transfer DNA contains genes which induces the formation. Can you see? Transfer DNA is the one. Wait. Is the one that contains genes which induces the formation of tumor. The mastermind behind the tumor formation. Right? Can you see? So, where the structure? See here. Now this is the thing. It actually happens. Now think what actually happens now. This is coming here, and the yellow color transfer DNA contains genes which will be genetically right. Sorry, the yellow color one is the transfer DNA. It contains genes which induces first. As soon as we want tumor form, it can't form right. First, it needs a starting point. So what starts that? What is the one that induces? Now we have to form tumor cells. We have to form the tumor. So, all right. Now this is the thing. Now this red color part is the agrobacterium, right? It has come to the plant stem and it has to form a tumor to survive without being exposed to external environment, and it should be in contact with the plant. So the only way it has to form the tumor. We know that the green color segment, this is the segment of DNA. This is the one that is genetically transformed into the cells of the tumor, right? But what induces now tumor? As soon as we want to form a tumor, it cannot be formed. First, it needs a inducing point. It needs a start, right? Which starts that? What is the thing that induces the tumor formation? See here. What induces? No, see here this last point. See the last point. Transfer DNA. So most of us confuse this. See here, first initially we knew that say cells of the tumor are genetically transformed by a segment of DNA. Last minute, see the green color part, right? But we thought that no, sir, okay, it was a bit confusing. But actually, what happens? Think like this. It is a bit easy. If you say like this, these genes in the transfer DNA induces the tumor formation. So this segment of DNA will be forming into tumor cells. Is that clear? Is that clear? Clear, right? Now transfer DNA is the one that starts. It says you start off. Now can you remember DNA polymerase? Now DNA polymerase, when I started DNA replication part, I said that DNA polymerase has the ability of polymerizing, but it can't start. To start, a primer is required, right? So similarly, here also it can form this green color. This segment of DNA can be genetically transformed into a cells of the tumor. But who starts it? Who induces? Who is the one inducing that? Transfer DNA. Is that point clear? Yes. After the inducing, this will be genetically transformed into the cells, right? And then the cells will be formed on like this, right? It will be forming back to back cells, and this is the tumor. Clear, right? Is the voice lagging? No, right. Okay, now this green color is the tumor, and these small structures are the tumor cells, right? Okay. Now we said that transfer DNA is the one that is transferred into the plant genome. Can you remember? First I said like this. The plant genome is present like this. This yellow color part is the one that is transferred into the plant genome, right? Clear? Right. Until this point, we didn't find any human interventions or human involvement, right? So what now we do is scientists said that
Why should we need these virulent genes? Is it benefit? Is there any benefit having virulent genes? No. What does virulent genes cause? They'll be causing diseases. They'll be causing inducing the pathogenicity. So only we have disadvantages. So they said that what if we remove that virulent genes and there's a space created, right? In that space, what if we inserted the DNA of interest, the DNA we want? Can you understand? What was their plan? They said that virulent genes, even if they have the virulent genes, there is no purpose. It has only a disadvantage. It affects the plants. So, what if we remove that virulent gene and create a space for the DNA we want to be inserted? Can you see? It was a mastermind plan, right? There's two benefit from this. One, the plant won't get affected. Second one, we can fulfill our need too. Can you see? Yes. So what they did, they slowly removed this. What did they remove? What did they remove? Virulent gene. They removed the virulent gene and created a space. And and that space, what they inserted was they inserted the DNA of interest, the DNA which we wanted. Ah, okay. DNA of interest is interest. Now, but ask why did they insert the DNA of interest here? Because see here, what happens? They said that transfer DNA is the one that is transferred into the plant genome. Can you see? So, as this transfer DNA now has the DNA of interest which we wanted, now that DNA also will be going into the plant genome. Will you agree with that or not? Yes. So, from this, can you understand that we can transfer DNA through this method also? But this is a bit longer. This is a bit longer compared to others, but this is also a good method. Can you understand? Yes. Now tell me, when the transfer DNA now is transferred into the plant genome, will the plant, now this is the plant stem, right? Will the plant get affected by a disease? What happens to the tumor cells afterwards? Tumor cells remains, tumor cells will be remaining in the outer part, right? It will be remaining. What we want is the tumor cells are now we know from the knowledge of you mean what you're asking, what happens to these tumor cells, these green color ones, after this transfer DNA goes, right? Yes. Now this transfer DNA will be going. Now these cells are formed from this, right? So after transfer DNA is gone to this part, they won't be further inducing the tumor formation. The more production of cells will be stopped because transfer DNA is the one that is inducing, but the already formed cells will be remaining. Is that clear? Yes. Okay. Right. Uh, right. Now, when this transfer DNA is transferred to the plant genome, which contains the DNA of interest here, tell me whether the plant will get any disease or will it get affected? Yes or no? No. Why? Why? You don't hesitate to answer. I know you know the answer. No. The DNA of interest has no harm for the plant. Correct. Yes. But I expect another term. See here. Here, before inserting the DNA of interest here, before inserting the DNA of interest here, if we transfer this transfer DNA, will it cause any disease? Yes. Why? It contains virulent genes. Can you remember? Can you remember? So what we did, we removed those virulent genes and inserted the DNA of interest. What you said was correct. The DNA of interest has no effect. But initially at that location, what was present? Virulent gene. That one is a problem. You didn't see the diagram, right? This diagram, right? Is that clear? Yes. Now, can you see here? Initially, there was a virulent gene, and now we have removed that and created a space, and we have inserted DNA of interest. Now here, can you see we have made a change in the transfer DNA? Can you see? Yes. For this we use a term. This process. Simply, this is the for this we say dis disarming the transfer DNA. Disarming the transfer DNA. So see, we have removed the virulent gene and added the DNA of interest. So this is called disarming the transfer DNA. Is that clear? Yes. Is it hard actually? I know it's complex, but it is. Is it hard? Yeah, it's a bit confusing. I know. Now, all right, right, fine, right. But tell me, from the four methods: transformation, transduction, gene gun, agrobacterium, which is interesting, which is engaging. This one, right? Even if it is a bit confusing, that made your attention to be, at one stretch, right? Those three were theory based. This was practical one, right? Okay, let's go to the note part, and if it's clear, we'll finish it off for today, right? Okay. Now, yeah, I have one. Yeah, this part, right? Okay, let's see. Therefore, scientists have removed most of the bacterial genes, including virulent genes from transfer DNA, and made room for the inserted genes of interest into the space between the two border sequences. Agrobacterium can deliver this modified transfer DNA with the insert gene to the plant cells through its infection ability. Infection ability means simply what is that? See here, it is inserting its DNA into the genome of plant, right? So that is called infection ability. So initially, it will be causing an infection, disease. Why? Because virulent genes are present. But now, even if it causes an infection ability, the plant won't get an infection. Why? 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 Now there is no infection caused. Agrobacterium transfers this white colored DNA of interest by its infection ability. But the plant is not getting affected. Why? Virulent gene is removed. Exactly. Right? Very good. However, plant cells will not become sick because the virulent genes have been removed from the DNA, transfer DNA. This is called disarming the transfer DNA. Right? Now, finally, just go through this part and let me know if you have any doubt. The agrobacterium part. If it's completely clear, then that's it for the day. Right? Just go through. Now, after this class, after it finishes, you have two work to do. One, what is it? Try the DNA packaging short questions which I send, which I'll be sending by three, not three. After this class finishes, I'll send it. Right? First one, you try that. Second one, please revise this section. You know how much hard this is to understand, right? It is a bit confusing. So you can't keep on watching the recording again and again. So as soon as the class finishes, after some time, after eating, praying, you can just spend some time, not more than 1 hour. Just spend some 1 hour of time and just revise it. Just read it and try whether it clicks, whether you can understand those, right? Clear. Right. Then let's move to the next part. Right. Check this part also. Finally. Clear. Okay. Then. Clear, right? No doubts for today, right? DNA library, DNA delivery system. Two big topics we have done, right? No doubts in both, right? Both are clear, right? No doubts, right? Clear, right? Okay, then that's it for the day. Thank you for joining live, right? So make sure to revise it before next week's class. Next week, hopefully, inshallah, I'll keep a physical class, right? So revise it and come. I'll be giving some papers next week, right? So let's do some MCQ, some questions next week. So make sure to revise and come, right? And do the short questions which I sent, right? Okay, then that's it for the day. Thank you. Welcome.